bear with me here, Mr. Hayhurst. I hope I don't bounce around too much, but I will do my best to give you forewarning on where I'm going. Next record that I want to talk about with you is under your lab number GR162492, record number 1. I'm going to hand you what's been marked as People's Exhibit 170. First and foremost, is that a report that you authored? Oh, give me 1 second.
Yes, it is. People's Exhibit number 170 is a copy of my laboratory report for GR16-2492. It is record number 1, and it is a DNA report. It's a complete copy of the report, and the second page has my signature at the bottom.
Okay. And in particular, you were working off of somebody had actually done some swabs for you, and this was the work as a result of that. And the swabs were from what?
So 2 items were either submitted or they were swabbed in the laboratory. I have to look. But they were swabs of they were identified as being swabs of a .22 caliber round. So one swab of the first .22 caliber round and then a second swab of the other one were submitted as 2 separate items for DNA analysis. And
these, and whether or not you know this, just for the record purposes, these were the 2 rounds found on North Green Creek Road after Madison Nygaard was from the car Madison Nygaard, in the area where Madison Nygaard jumped from a car.
Okay.
Now, your work, excuse me, as it relates to this, were you able to come to any conclusions?
Yes. So, excuse me. The 2 items underwent the normal DNA analysis process as 2 separate items, and I received DNA results from each of these items, and they're reported on the bottom of page 1 of my report.
Okay. What conclusion did you come to?
So the first item was identified as item 1AX in the analysis, and that's that swab of the first .22 caliber round. It did give a partial DNA profile, but it was a partial enough profile. There was not enough information present, and so I deemed it insufficient for comparison purposes. In a sense, it's inconclusive. There's just not enough data present for me to make any comparisons.
Okay. And the second item?
So the second item, again, looking at the data, it was clear to me that it was a mixture of multiple contributors, and this is a scenario, one of the scenarios where it fits into that idea of a complex mixture. It was not something where I could easily pick a major and a minor, and so at this time frame, in April of 2016, was shortly after we brought our first 2 scientists online with using the StarMix software in our laboratory. Because I was not a part of that first group, a sample such as this, what I would do is I would provide the electronic data to the analyst who actually would then run the comparison, run the analysis, and do comparisons that were applicable. So the conclusion here is just that it's a mixture and that further analysis will be the subject of a separate report.
Obviously, going back to some of your initial foundational testimony, again, we're talking about 2 items where not so much quantity, but we're looking for quality. Is that?
Actually, it could be both. Likely quality based on the information I had as far as recovery location, being outside in an environment, whether it's the light or the heat and humidity, whether it's the dirt from the ground that could inhibit DNA. There's a variety of different things quality-wise that apply. But also, I would say, I would offer on items such as this, quantity can sometimes also be a concern. It's not an item that you are frequently going to touch. A lot of times with a .22 caliber round like this, you take it out of the box and you load a firearm, and that might be the only time you touch that. If you unload, maybe load it again, you're really talking very limited and not a lot of contact that's repeated that's going to leave a good source of DNA behind.
All right. And I think you indicated your signature's on the bottom. Anything different about that report than the one that you originally authored and submitted?
No, there's not.
Your Honor, I move for the admission of Peoples 170.
What number is that?
170?
James? Any objection?
No, sir.
Case received.
Okay. Under that same lab number, we're going to move to record number 4. That's marked as People's Exhibit 171.
Yes, People's Exhibit 171 is a copy of my record number 4 report for this laboratory number. It's a total of 7 pages, and it does look intact. It has my signature on the bottom of the last page.
And how would we classify this report? Is it a serology or a DNA report?
This would be along the lines of the serology report, which again is that initial screening of the evidence for possible biological value, trying to determine which samples to forward for DNA analysis. This is that initial screening.
Okay. So I brought up to the front there, this is People's Exhibit 126. I see you brought your own gloves. I'm going to have you go ahead and, I guess, glove up there, and we're going to go through a couple items here.
Trying to figure out the best way to do this.
I'll move this out of the way for you. All right. You all set?
Yep.
If you don't mind, just so that I can kind of work through this report, I'm going to sit because I've got it on my computer screen, so hopefully that doesn't offend anybody. Let's talk about, in that record number, I want to talk about item L6 for a moment.
Okay.
And item L6 is what?
So item L6 was one of the items that was contained originally within the outer toolboxes. So item L6 is a pair of 2 black Reebok gloves.
Now, this is probably a good spot to just talk briefly when you do this serology examination. What are you looking for?
So again, the serology exam is an effort for myself to look at the evidence that's provided and determine potential sources of biological value. Again, the easiest would be a visible stain that's present on an item. So on most of these items, a bright light examination for potential discolorations or stains that could be tested for blood. But then also after kind of that cursory look to see stains that might be present, then considering other types of DNA that may be present on an item, again, such as the contact DNA type example we talked about earlier.
Okay, so in this particular instance, in referencing this report, what tests did you perform and did it provide any information to you?
So specific to item L6, and it's actually these right here, it's a apparent pair of Reebok, like a baseball batter's type style gloves. I did note that they were significantly worn. But then the initial examination was to visually examine and see if there's any potential blood stains that are present. On this item, there were a few areas of discoloration, so I actually tested, I think, 3 separate areas. I have a chemical test. Several chemical tests are available to test for blood. And the initial, on page 3 of my report, about halfway down, item L6, the initial test that I conducted because of the nature of this evidence was a phenolphthalein test, and it's a color change test. If there's a color change, then it indicates the possible presence of blood. And one of the stains on the exterior of this glove, or one of the discolorations on the exterior of this glove, did give a positive phenolphthalein, which is an indication of the possible presence of blood. I like to follow that up with a hemotrace test. It's a slightly different style test. It's more of an immunological test that's specific for proteins in human blood. And that test did come back as negative. The hemotrace was negative, so it did not indicate the presence of human blood. Whatever stain may have been present could have been a variety of other things, but it did not initially indicate positive for that. Regardless, again, then you fall back to the secondary position of what other potential sources of DNA on this glove. And so actually at the time of this report in May of 2016, I did go ahead and collect a swab of the interior of the glove. And again, that's the idea of trying to pick up DNA from somebody who would have been wearing the glove and depositing their DNA on the inside of the glove. So I did collect a swab at that time. It was just not run for DNA analysis until a later date.
So just so and I thank you for referencing the date because I forgot to do that. This report indicates you are doing this lab work in May of 2016.
That's correct.
Okay. Now, going back to your hemotrace test, is that test affected by the quantity of the stain that it's tested?
Yeah, that's a very safe assessment. The hemotrace test, as compared to the phenolphthalein or hemostix test I also have available, the hemotrace test is much more specific for human blood, so I do like to do that. However, it takes a little more sample. It's not quite as sensitive as that initial test, the phenolphthalein test. So I might have a very dilute stain that's strong enough to bring a positive result on the initial test, but maybe not enough of the stain is present, or I didn't swab enough for the actual testing itself. So is that a possibility? Yeah, if it's a very dilute stain, then it would not, it might not be enough where it's able to give me the positive hemotrace result. We'll still report it as a negative test. It did not indicate the presence of human blood, but that doesn't mean that maybe down the road we wouldn't run that sample for DNA analysis. And I've had a number of other cases where maybe there's that feeling that it was just not a strong enough sample to give that positive test at this stage. And let's go ahead and forward it for DNA analysis because that's a very sensitive method anyway.
Okay. So, and you did indicate that you did swab the interior portion of those gloves at this time.
That's correct. In May of 2016, I swabbed the interior portion of the gloves.
Now, storing those swabs, and I'm going to move this podium because I can't see you. Storing those swabs, how is that done?
Yeah, so initially in May of 2016, I knew this entire container of evidence was going to travel to different units in the laboratory. And so I wanted to collect as many samples initially as possible. So a variety of items in here were swabbed, and the process is just using like a cotton Q-tip type swab. We use a sterile cotton swab and sterile water. Swab the stain or the area of interest. That swab is dried, and then the cotton portion I actually will cut off and I'll place into a small DNA extraction tube. That tube goes into an envelope, and then all the samples from a case go into the same envelope. That's what's then forwarded for DNA analysis. That way, when I get up to the DNA laboratory upstairs at our laboratory, I don't have to deal with a large pile of evidence. Everything is in a tube, ready to go. I just have to add chemicals. So at the time, the swab was collected, dried, cut into a tube, and sealed, and it remained sealed until I actually ran that sample for DNA analysis at a later date.
All right, let's talk about item L8, which is the mouth ball gag. Can you describe what you did with that, please?
Yeah, so item L8, and all of the items in here I've placed a laboratory sticker with my signature, or my initials, and our laboratory information. This item was an apparent ball gag. And initially, did the visual examination to determine if there were any blood stains that were present. I did not see any stains, so there was no stains for testing. And then again, knowing that this evidence was going to travel elsewhere in the laboratory, I tried to consider different areas on this that may be potential sources for DNA. So initially I swabbed the actual red ball itself as a sample for DNA. And I also believe at this time frame I swabbed the leather straps that would have contact with the skin of whoever may have this item.
All right. Moving on to item L10.
Item L10, again, has the laboratory sticker and my initials, was a purple vibrator dildo that was submitted within the outer container.
And did you do any swabbing of that particular item at that time?
Yeah, so again, initially did a visual examination to see if any potential blood stains were present. Did not note any. Based on previous trainings that I've had, I did note that at least some of these surfaces may have the potential for latent print analysis. We're always worried about trying to preserve evidence for other units in the laboratory. So I actually had a consultation with one of our latent print examiners at the laboratory, and she indicated that the lower portion could be potentially a good source for fingerprints, but that the upper portion was not a good printable type surface. So at this time, in the absence of any visual stains, I swabbed the tip of the dildo vibrator, and same process. Swabbed it, dried it, cut it into a tube, and then sealed that.
All right. Item 11, I guess what's labeled as the silver vibrator.
Item 11, again, my laboratory sticker is on here. It was an apparent silver vibrator with the attached battery pack. Initially, there was a stain on the, I believe it was the silver end. There was a possible a discoloration, a possible stain for testing. So my report does reflect that I did some hemostix test on this. Again, that's a chemical test, indicates the possible presence of blood. And in this case, it was negative. It did not indicate the presence of blood. And then, again, thinking ahead towards what are some potential sources of DNA on this item, I did swab the silver portion of the silver vibrator, prepared that sample for DNA analysis.
All right. I took it away from me. Let's talk about the work you did on People's 127 real quick. The handgun. Did you get an opportunity to also work with that as well?
So People's Exhibit 127 is a box, again contains some laboratory stickers. Contained within is what we have identified as item L38, and it is a handgun that was recovered as a part of a crime scene investigation.
And again, the analysis you went through, observation and swabbing, could you describe to the jury, please?
Yeah, so again, initially the examination was for the possible presence of blood, so a visual examination was conducted. I did note that there was some small, it had some various debris and dirt, as you would expect might collect on an item, but I did note on the end of the muzzle, my gun terminology is not precise, but on the end of the muzzle, the barrel end, there was some discoloration that was noted, and so I did test that. I used the hemostix test, and it was a positive result, so it did indicate the possible presence of blood along this, the outer end of this, and it was actually when I received it, this gun was still in this format, and I could see the outer, the staining was visible along the exterior here. So I collected a sample for DNA analysis, and on page 6 of my report, L38 indicates the positive hemostix result. I did swab that area. Also at the same time, I swabbed the grip and the trigger, again thinking of what are some possible areas for contact, how would an individual interact with this. So I swabbed both the trigger and the grip areas, and that was a separate DNA sample. And then this gun actually traveled through the other units of the laboratory, went to our latent prints unit and our firearms unit. When it was in the firearms unit, our firearms examiner, as a part of their examination, actually had opportunity to take the gun apart in ways that I don't even understand. He took the top portion, the slide, as I think of it, and actually exposed this inner barrel. And I was able to see there were some additional stains that I did not initially see because they were in a protected area. So I collected a third sample from this, L38, I believe C is the third sample that I collected. And they were, again, some stains that were present on that interior portions of this handgun.
Okay. And I'm not going to have you keep repeating the process, but we're talking about that process where you swab, dry, tube, and set aside until you can run the analysis.
Correct.
Okay. Now, going back here to record number 4, also, you closed up the box a little quick on me there,
Yep.
Mr. Hayhurst. But going back up the report to, let's talk about L16, the J-shaped hook with the ball on it.
Item L16, my laboratory sticker, is a large J-shaped hook with an eyelet on the end and a ball on the opposite end.
And what did you do with that?
So initially, again, the visual examination, looking for the presence of possible blood stains on this item. There was at least one area that did have some discoloration, so I did follow up with a phenolphthalein test on this, and it was a negative. It did not indicate the possible presence of blood. Once the screening for blood is done, then trying to figure out what areas might an individual have contact with this item. At that time, I did swab the ball end, sterile cotton swab, sterile water, cut it and prepared it for DNA analysis on that ball end.
All right. Excuse me. Moving up to L14, the length of chain with attached handcuffs. What did you do with that?
Item L14 is a length of chain, approximately, I think it was a little over 3 feet, and then actually had a pair of handcuffs that was attached to that last link. This was contained within this outer toolbox. It does bear my laboratory sticker and my initials. L14 originally examined with bright light for the possible presence of blood. There was a few areas, there's some areas of like rust that are noted here, so that was kind of taken into consideration. There were a few areas that quite possibly could have been rust, but in an abundance of caution, I went ahead and tested a few of those areas, and then a few areas of discolorations. Used the phenolphthalein test, and in each of these areas, the phenolphthalein ended up being negative. It did not indicate the possible presence of blood. I did swab at this time in May of 2016. I did swab the interior portions of the handcuff. Again, the area where an individual may have contact, skin contact with this item, and so that swab was taken at this time and forwarded for DNA, or prepared for DNA analysis.
Okay. There's also, as well as the Reebok gloves, there was a single glove too. That's L17. Is that correct?
That's correct. L17 is a single black glove, again, an apparent, like a baseball batter style glove. This was a Mizuno branded glove, and it was a single. It is the right hand.
And what did you do with that?
Again, initially looking at looking at the surface with bright light, trying to identify any potential stains, any areas of discoloration for testing. There was a small area that did have some discoloration, so I did test it for using the phenolphthalein test, and it was negative. It did not indicate the possible presence of blood. Fall back to that secondary. How else could people potentially have contact with this? And again, at this time, I swabbed the interior of this glove. Again, the palm area where an individual may have had contact with this glove. And in May 16, that is the area that I focused on.
And the last item that we're going to talk about in this box is L18, the leather handcuff restraint with connecting metal rod.
Excuse me. Item L18 is a pair of apparent leather restraints that are connected by a chain, and then the chain has a rod to hold them separate. This item was examined. Again, examined with bright light for the possible presence of bloodstains. I did note a few areas for testing. However, in each of those areas, the phenolphthalein test that I conducted was negative, and so it did not indicate the possible presence of blood. Then again, fall back to the second question of what areas might an individual have contact with this item, where could I potentially find skin cells from that contact. And so I did swab at this time the interior of each of these leather restraint areas, again with the idea of where they would have had contact with an individual. That swab was prepared for DNA analysis.
Now, I'm not having you go through everything, but obviously there are some other items that you did similar examinations to, as you've talked about. Is that correct?
That's correct.
And all that information is contained in that record number 4?
Yes, it is.
And again, this is all being prepared and done in May of 2016. Yeah,
initially the first time I seen this, I examined this toolbox in the laboratory was in May of 2016.
Now, you were, and correct me if I'm wrong, but you were part of the mobile crime unit, is that correct?
Yes, on this incident, when this was collected several days prior to this, I was actually a part of the crime scene response team that went out and actually recovered this toolbox. I actually recall that I even was the one who used our bolt cutters to snap the lock when I was at the scene.
Okay, so you see this toolbox being recovered from the van, the silver van, is that correct?
Correct.
Obviously, it's packaged up at that location and then forwarded on for you to do your work that you just described.
That's correct.
Okay. Your Honor, I move for the admission of People's Exhibit 171, record number 4. No objection. And I can't remember, did I move for 170 as well or no? I think I did.
Yes, you did.
Okay.
170 is in.
Okay. Record 4 is 171.
Any objection to 171?
No, sir.
That'll be received.
Just a second. Might as well keep those on.
Swap it and pair out.
Okay. Okay. Do you?
Sorry, I'm good. Those are a little smaller than I.
That's okay. So I'm handing you what has been marked, or what is, I should say, People's 138, and I believe it's your report associated with that, and that's record, or excuse me, People's 172, which is record number 7 of that same GR16 complaint number.
Yes, People's Exhibit 138 is a brown paper bag that bears laboratory stickers, other identifying information, containing several items of evidence that I examined. And 172 is a copy of a laboratory report that I issued in this case. It's a 2-page report. It is complete. My signature is on the bottom of the second page.
Do one other thing here. Also handing you what's been marked as People's Exhibit 173. Your Honor, it's my understanding that the defense counsel and I are stipulating that the known samples belonging to the defendant, Jeffrey Willis, come from that buccal swab, and there's no dispute over that.
So are we admitting Exhibit 173 then?
Yes, sir.
Okay. Now, you've not offered 138 or 172, you've just shown them to the witness.
Well, 138's already been admitted,
Oh,
I think.
Well, 172.
172 I've shown. Okay. Let's talk about what you did here, or what you received, I guess, what this report is telling us, Record 7 is telling us.
Okay.
You received, what's been stipulated to, you received a known sample, what we typically call a buccal swab, which I think you've kind of identified from the defendant, Jeffrey Willis. Is that correct?
That's correct. It contains a buccal swab. Again, that's a swab of the buccal cells on the interior of the cheek. We like those for reference samples because they're pretty non-invasive, and it's a good wealth of DNA. So that was collected and submitted under this laboratory report.
Okay. And the other item, which is the People's Exhibit 138, was also received via this lab report as well. Is that correct?
That's correct. They were both submitted to laboratory at the same time, and so because they were submitted at the same time, they are both reported on the same report.
And again, this report, Record 7, is this a serology report or a DNA report?
This is the serology type of report that we have. Again, that's the initial screening for the possible presence of stains and DNA on an item.
And did you perform on those two pairs of underwear any sort of tests?
Yeah, so I received 2 pairs of underwear. One is a pink pair of panties and one is a red pair of panties. Again, any evidence kind of gets that cursory look, or their potential for blood stains that are present. But specifically for an item such as a pair of panties, I did some examinations to look for the possible presence of sperm cells. So the pink pair of panties was first, and I examined them. There is a small area, and I did cut the sides in order to access them in the laboratory. It's easier for testing. There's a small area in the crotch of the panties, and you can see where I have removed a cutting. There was a small stain that was present there. I have a chemical test that indicated the possible presence of seminal fluid, in that area. And then I followed up with a microscopic examination. I prepared a slide, and it's actually in this little holder here. Prepared a slide and was able to visualize some actual sperm cells that were present. So the combination of those tests did confirm the presence of semen in the crotch panel of those panties. And so a cutting was taken from that crotch area of the panties for some DNA analysis.
All right. And did you do a similar similar examination as far as the red panties were concerned?
Yeah, so the red panties, again, were examined in a very similar type of a fashion. In this case, there was no, some AP testing, some acid phosphatase testing, did not indicate, it was negative. It did not indicate the possible presence of seminal fluid. So instead, with this pair of panties, I actually took 2 samples, both of them contact type samples, and I actually believe only one of them ended up getting run for DNA analysis. Specifically looking to who might be the wearer of this pair of panties. So I swabbed the waistband, the interior of the waistband of the panties, cut that swab into a tube, and prepared that for DNA analysis.
And essentially, is that, I mean, ultimately, is that what Record 7 tells us?
That is correct.
All right. Again, anything different about that particular, what's marked as People's Exhibit 171 than your original report? Or excuse me, 172?
172 does appear as I issued it.
All right. All right, move for the admission of People's 172.
No objection, Your Honor. No objection? That's 172 is received.
Okay. Yes, sir. Okay, handing you what's been marked as People's Exhibit 174, record number 8 of that same GR16 laboratory number. What, again, is this a serology report or DNA report?
People's Exhibit 174 is a copy of a laboratory report that I issued, and this is a DNA formatted report. Indicates the samples that were actually submitted and tested for DNA analysis.
Now, does it indicate the evidence that you were testing? Let's start with the known samples that you had.
Yeah, so on the top of page, or in the middle of page 1, is the beginning of the listing of all the evidence that was available to me for testing and comparisons. The first two items, the alternate reference from Jessica Herringa, and then the DNA profile from Rebecca Bletch. Both of those were, the data for each of those items was actually generated in the laboratory previously as a part of those other associated cases. I was able to use the data from those other cases. I did not have to repeat that analysis because we'd get the same, on a reference sample you're going to get the same result. So I was able to take that electronic data from each of those cases and have it available for comparisons. Additionally, the first two items that are listed that are new on this report would be that DNA swab, the DNA profile from Jeffrey Willis, and the DNA profile from Madison Nygaard.
Okay. Then, as you scroll down the list, it also includes the swabs that were done in that serology report. Some of which we went through, and some of which we didn't go through, but they are the information or the process, the DNA results are part of this report. Is that correct?
That is correct.
Okay. So let's, and also maybe can you explain, so you've got this list of the swabs, and then after the last stain and crotch mark, you have kind of a longer list. Can you explain to the jury what that means?
Yeah, so again, initially knowing that this evidence was going to travel to the laboratory, I wanted to collect as many samples as I thought may have the potential for going for DNA analysis at some point. And so I actually collected a large number of swabs from this item, from the toolbox, and prepared them for DNA. Only a selected number of those items actually were run for DNA analysis on the first round through. And so that's where the bottom of page 1 and the top of page 2 list the items that I actually did conduct DNA analysis on initially. That middle section on page 2 actually is the list of all of the swabs that were available to me, but based on time and resources and investigative avenues at that point, those samples were not run for DNA analysis at that point. Some of them were actually run at a later date, some of them have never been run and still exist in the swab form in a tube. But that's the sum total of all of the rest of the samples that I had initially collected.
So this might be a good point, Mr. Hayhurst, to talk or ask you questions about, we had some testimony from one of your counterparts who works in the firearms unit that your lab services 17 counties.
Our laboratory, that's the actual number of counties I would not remember exactly. Actually, for DNA analysis, the Grand Rapids Laboratory primarily is responsible for servicing all of West Michigan, so say Ludington all the way to the Indiana state line. But then we also actually do DNA analysis for all of northern Michigan, so everybody up to the bridge, and then all of the UP. All of the DNA samples from around the state, the different labs, come to our laboratory from the Grayling and Marquette labs and our own. So we, actually for DNA analysis, we cover somewhere, I would guess, 30 to 40 counties, potentially.
And thank you for clarifying that, because I didn't realize that.
Yeah, we have a lot.
So as a result of, and obviously you're dealing with all the, not only the state police posts that service those areas, but also the local jurisdictions as well.
Correct.
So that's a lot of police agencies.
That's correct.
Now, as a result, you talked about resources and time. I'll apologize up front. I can't remember. You talked about the process, but did you talk about the length of time from start to finish? What does that take?
Yeah, so the initial screening of a piece of evidence, if it's a single piece of evidence, doesn't take long to look for bloodstains and swab for DNA. That can be conducted in a matter of hours. But the DNA analysis process, because it's multi-step, because there are times that are built in for each of those different steps, there's some interpretation time that's built in, the review time. To actually conduct DNA analysis on an item, it's pretty common that you're looking at a backlog or a turnaround time of several months between when an item is submitted to the laboratory and when you might get that first round of results.
And as a result, does the state police have a policy as it relates to the number of items an agency can submit and I guess the priority of what is tested?
Yeah, so we have a policy that generally indicates we are to limit it to 7 samples being actually run for DNA analysis initially, and then whatever reference samples are available, we don't count those against. But as far as evidentiary type samples, we try to limit to 7 samples for homicide type cases. For all other cases, we generally will limit agencies to 2 DNA samples initially. So again, that's just an effort to have us try to stay ahead of not having huge backlogs where we increase turnaround time. So we do have some guidelines for how many are tested.
And the one thing you did mention, because you're currently doing it, you have to build in for court time too.
That's correct.
I mean, you get subpoenaed because of the work you do.
That's correct.
Okay.
Frequently far away. Last 2 weeks ago I was in Munising, so it's not necessarily local testimony either.
Correct. Okay. So that does that put in context a little bit, Mr. Hayhurst, you know, what when we talk about these lists and what was and wasn't done, essentially that's what agencies are dealing with and you are dealing with based on resources.
That's correct.
Okay. So let's talk about let's talk about the process that you used in obtaining the DNA results for the particular pieces that you're looking at in this record.
Okay.
What process were you using?
So again, at the time frame in May of 2016, the DNA analysis that we were conducting in the laboratory was specific to a specific chemistry that's known as PowerPlex fusion. There are several commercially available DNA analysis kits that are out there. There's primarily 2 at this time frame. And at the time of this analysis, I was using PowerPlex fusion. There's a note above, on page 2, just above where it says PowerPlex fusion, 2 of the reference samples were actually run in the last generation of that kit, of that technology. It was called PowerPlex 16 Hot Start. So 2 of those references, when the time they were analyzed in the laboratory, we were still on the older kit. That looked at 16 different locations. This next generation kit actually looks at 24 different locations. So it's a more informative, and it's actually quite a bit more sensitive kit. So all of the testing that was done on this evidence was done with the fusion system, but then they are compatible. So you can do comparisons to older reference samples.
All right. Let's start on page 3 here. And had you run through your results. The first result had to be a swab of the passenger side door lock, which I'm assuming was from the van the silver minivan that you were there collecting evidence from?
That is correct. I collected this when we were on the scene. It was a swab of the door lock of the passenger side of the van, the front seat passenger. DNA results obtained indicate that it was a mixture of multiple contributors. It was a complex mixture, so this was one of the samples that I elected to forward for that StarMix analysis, the computer modeling that was done.
Okay. Obviously, those results are not, in fact, I think you referenced the record where those are located.
That's correct. They were conducted at a later time by another analyst.
All right. Let's look at your analysis of the swab of the red ball on the ball gag. What were you able to determine?
So the DNA profile from the swab of the red ball and the ball gag was a partial DNA profile. It was a single source profile. It's from a single individual. It's not a mixture. And it was from a male contributor. That partial DNA profile matched the DNA profile that was obtained from Jeffrey Willis. The two profiles were correspondent. Again, anytime there is an inclusionary type statement like that, we provide a statistic. And so this will be statistic number 1.
Let me get there.
Yep.
All right.
Statistic number 1 is on page 6. And again, this is the probability of selecting another individual random who also matches that partial DNA profile. It would be 1 in 50.48 sextillion in a Caucasian population, 1 in 7.886 septillion in an African American population, and 1 in 1.036 septillion in a Hispanic population.
Okay. And then the other conclusions, let me get back to that. The other reference samples that you had were what?
Yeah, so part of our policy is that we have to compare all of the references to an item of evidence, even if we have already established some kind of an association. So in this case, Madison Nygaard, the alternate reference from Jessica Herringa, and Rebecca Bletch are all excluded as being possible donors to that red ball on the ball gag.
All right. The next item was the swab of the end of the dildo vibrator. I'm assuming that's the purple one, if we're referencing between the two?
That's correct. It was the tip of that purple and blue dildo vibrator.
And what did you find there?
Again, that was it did indicate the presence of a mixture of multiple contributors. Because of the nature of the mixture, I elected to forward that sample for comparisons using the StarMix analysis. So that was provided, that data was provided to another analyst.
All right. The silver end of this would be the second vibrator. What were you able to determine there?
That's correct. The silver swab of the silver end of the vibrator did give a partial DNA profile, so I didn't have data at all the locations. But the DNA types were consistent with being a mixture of two individuals, so there was a mixture. But this is an instance where one of the donors was present in a more excessive amount, so we have a major donor. And at this time, that was identified as being a major unknown contributor. The major donor is from an unknown donor, so I called it unknown donor number 1. And then likewise, as it is unidentified, Jeffrey Willis, Madison Nygaard, and Jessica Herringa and Rebecca Bletch are all excluded as being possible as that major donor from the swab of the tip of the vibrator.
All right. In the minor donor analysis?
So the minor donor was at a level that was low enough. It was not a lot of data from that secondary donor. I could tell there was a second donor there, but it was a low enough level that it was deemed insufficient for conclusive association purposes. Just can't make any comparisons.
So the quality of the DNA wasn't sufficient for you to make any kind of analysis or match, if you will?
Either the quality or potentially even the quantity. There's not, comparatively speaking, as much DNA from that second donor, so I'm not able to make a comparison.
All right. Next, your analysis of the interior of those leather restraints. And I believe that was the one where it had the steel bar in between.
That's correct. The leather restraints with the steel bar, the chain.
All right. What did you find there?
That was item L18, and the swab was L18AX. That DNA did result in data that was consistent with being a mixture of two individuals. Again, this was an instance where there is a more major contributor, and it was a male, the major male contributor, and then there's a minor contributor. The minor is low enough that it's insufficient for comparisons. That major donor, though, was compared to the available reference samples. And the DNA profile from Jeffrey Willis matches the major donor from the swab of the interior of that leather restraints. Again, because there's that association, there's a statistic, number 2, on page 6. Probability of finding another person in the population at random who also has that DNA profile would be 1 in 1.796 septillion in a Caucasian population, 1 in 1.220 octillion in an African American population, and 1 in 220.1 septillion in a Hispanic population.
Right.
Again, the other reference samples that were submitted were also compared against that major donor. Madison Nygaard, Jessica Herringa, and Rebecca Bletcher all excluded as being the major donor from that swab of the interior of the leather restraints.
Okay. Next was your analysis of the stain, swab of the stain on the muzzle end. So I know you described finding 2 stains, one on that muzzle end and the other on the interior. This is the one that was exposed, I guess. Is that a good way to describe it?
Yeah, that's fair enough. L38AX was the swab of the stain that was on the end, that portion that was visible to me initially before the gun was disassembled.
Okay, and what did you discover?
Partial DNA profile, partial DNA types, so it was not a complete profile. However, it was consistent with being a mixture of 2 donors. There was a major male contributor. That major male donor was compared, and the major DNA profile from Jeffrey Willis matches the major donor from the swab on the stain of the muzzle end of the .22 handgun. There's the association, so the statistic is provided. It's number 3 on page 6. Probability of selecting another individual random who also matches would be 1 in 1.935 quadrillion in a Caucasian population, 1 in 14.01 quadrillion in an African American population, 1 in 10.43 quadrillion in a Hispanic population.
Right. And it appears as though you also ran the swab of that handgun, the interior swab that you had described underneath the, or hidden, I guess, by the handgun slide.
That's correct. That was identified as being item L38CX. And that was the one that was on the interior surface.
And what were you able to determine there?
Excuse me. Again, DNA types are consistent with being a mixture of at least 2 individuals. This was another instance where there is a more major contributor and a minor contributor. Jeffrey Willis, the DNA profile from Jeffrey Willis matches the major donor that was obtained from the swab of that interior end surface. There's a statistic that's associated. This one's number 4. Probability of selecting another individual from population who also would match that would be 1 in 192.4 septillion in a Caucasian population, 1 in 282.4 octillion in an African American population, and 1 in 15.68 octillion in a Hispanic population.
Now you also, per your policy, ran it against the other knowns you had, is that correct?
That's correct. Each of the other knowns that were available at this time are all excluded as being the major donor.
Now, and again, in this particular instance, there was a minor donor discovered, and what was the conclusion of that?
Again, on this instance, the minor donor, the DNA types from the minor donor are low enough. They are insufficient that I can do any kind of a meaningful comparison. So it's insufficient for conclusive association purposes, and in a sense, it's inconclusive for that minor donor.
All right. Now, there's also, the next one is a swab of the van cargo floor, and I'm assuming you performed that swab as well, since you were again at the garage, I guess, where the van was held. Is that correct?
That is correct.
Okay. And that particular analysis did what?
The DNA analysis from that swab of the van area, the cargo area of the van floor, did give results that indicated the presence of a mixture of multiple contributors. And again, this was an instance where I forwarded that data for StarMix analysis and comparisons.
Okay. We move now to, it looks like the swab that you did of the interior of the red panties.
Yes, item 4E1X was, again, that swab of the waistband area of the red panties.
And what, if anything, were you able to conclude from that analysis?
So DNA types that I obtained, the data that I obtained from that item, are consistent with a mixture of at least 2 individuals. I could tell based on the data that at least one of those individuals was a male contributor, and there was one unknown contributor, so an unknown donor on that sample. The reference samples that I had available at this time, I compared, and Jeffrey Willis, the DNA profile from Jeffrey Willis is actually included as being a possible contributor to that mixture of DNA types from the swab of the waistband of the red panties. Because there's an inclusionary type statement, there is a statistic. This is number 5. And again, this is a little, it's on the top of page 7. This is a little different statistic than the random match. The random match is a determination of how rare that specific single profile is. Statistic number 5 here is what we call a reciprocal CPI. It's the combined probability of inclusion. Basically, you take all of the DNA types that are obtained in a mixture, and you can consider any number of pairings, and you determine how many other individuals from the population might also be included in this mixture of DNA types based on whatever pairings are possible. So it's actually not quite as informative of a statistic. It's maybe asking a slightly different question. But what it does is it gives a measure of how many other people could also be included in here. And it would indicate that in a Caucasian population, you might expect that you find another individual who's included in 1 in 930,200 individuals, 1 in 18,450,000 in an African American population, and 1 in 2,517,000 in a Hispanic population. So the numbers are lower, but we're asking a slightly different question as far as who else might have contributed based on the data that's obtained for that sample.
All right. And finally, well, I should say that the other 2 items, it appears as though they were F1 and F2, the 2 stains.
Yeah, so for the stain, that seminal fluid stain in the pink panties that was identified sperm cells, we have a slightly modified technique in the laboratory that we will use for seminal fluid stains. And what it is, it's a physical separation. Before I do any of the other DNA analysis process, we have some chemical physical separations where we attempt to separate any of the sperm cells from all of the other cells that might be present in a sample. That sperm cells is that F2 fraction, and then everything else is the F1 fraction. So we actually come up with 2 separate conclusions because after we split them, they're treated as 2 separate items.
And the F1 conclusion was what?
So the DNA profile obtained from that fraction 1 of the stain from the pink panties crotch was consistent with being a single source profile. It was from a female contributor, and at this point that's deemed unknown donor number 2. Jeffrey Willis, Madison Nygaard, Jessica Herringa, and Rebecca Bletch are all excluded as being possible contributors to that stain.
Okay, and I'm assuming then that analysis was saved for potential, I guess, comparison to any other known samples.
That's correct. At that time, that data was available for comparison against any additional suspects, victims, eliminations, anyone else who might have been a possible source. And so comparisons were done at a later date.
All right, and then your analysis on the F2?
Again, that fraction 2, the F2 of that stain from the crotch and the pink panties, did indicate the presence of a mixture of 2 individuals. At least one of them was a male contributor, and at least one of them was an unknown donor. And it's significant to see here that I have unknown donor number 2. It is the same unknown donor that we saw in the other fraction of the panties. The same unidentified profile was visible in both. In this case, they were compared against the knowns. Jeffrey Willis is included as a possible contributor to that mixture of DNA types from that sperm fraction of the panties. There's the statistic number 6. Again, it's that mixture statistic, so it's that idea of all of the possible combinations and pairings and who else could potentially be included. And you would expect that maybe you would see that in 1 in 222.2 thousand individuals in a Caucasian population, 1 in 102.8 thousand in an African American population, and 1 in 317.7 thousand in a Hispanic population.
Okay. And if memory serves me correctly, of the evidence, that was, well, let's go back and certainly it was, again, due to policy or because of policy you compared it to everything else, all the other knowns that you had?
Yeah, at this time, all of the loans that we had available that we believed could have possibly been donors were compared. So, again, on that stain from the crotch of the pink panties, Madison Nygaard, Jessica Herringa, and Rebecca Bletcher all excluded as possible donors.
And I believe that concluded your analysis for record 8. Is that correct?
That is correct.
Your Honor, I move for the admission of People's Exhibit 174, which is record 8.
No objection, Your Honor.
It'll be received.
Okay. Oh, that's fine.
Thank you, Mr. Hills.
Thank you. Okay. Handing you what's been marked as People's Exhibit 175H. Give me 1 second while I put that up on the screen here. That's record number 17, is that correct?
That is correct. People's Exhibit 175H is a copy of record number 17 in this case that was produced by the laboratory. This is a report that I produced, and it is 3 pages. It is intact and complete. My signature is on the bottom.
Now, this is a serology report, is that correct?
That is correct.
And in particular, what new did you receive, and what did you do in this, or what, I guess, the better way to ask the question is, what's this report telling us?
So after the initial round of DNA results were conducted, reviewed, released to the agency, an evaluation of all of the evidence in the case was underwent, and they determined they had some additional requests for DNA analysis. So I requested that they resubmit the toolbox, item L5, containing all of the items within, because I needed to do some additional work and swabbings for some of the specific items within the toolbox. And then also they resubmitted 2 separate containers. The first container, number 16, was a container that contained, it had a pair of black Adidas gloves, and then finally another container that had an additional reference sample that had become available for comparison. So 3 separate containers were submitted to the laboratory. Not all of the items got the full examinations again, had very targeted and specific requests out of what we wanted to see from here. But again, trying to get more samples to forward for DNA analysis.
And one of those samples was L6, that pair of black Reebok gloves, is that correct?
That is correct.
And I think in your initial analysis or initial work, you had indicated you had swabbed the interior of the gloves.
That is correct.
And in this particular report, what did you do with those gloves?
So this is, again, after the second time it was submitted to the laboratory. The specific request was for me to swab the exterior of the gloves. You know, commonly we want to see who's wearing them, but one of the questions arose would be who would have had contact with the gloves on the exterior surfaces. So item L6 was specifically focused on, and the request was made to swab the exterior of the gloves. Specifically, I swabbed the palm area and then the finger areas, because those would be the areas that you would have contact with another individual or another item, another potential source of DNA.
And is that true also for the single Mizuno glove, the L17?
That is correct. Very similar type of request. Initially, I'd swab the interior. This time around, they requested that I swab the exterior of that glove.
As well as I think you indicated you had received one of the newer pieces of evidence you'd received was a item L37, which was a pair, I should say, of black Adidas gloves.
That's correct. Item L37 was initially in another container, and on this item, I specifically was requested to swab the exterior of that apparent pair of Adidas gloves.
And I don't know, Mr. Hayhurst, if you recall, but was that pair of gloves also part of what was found in the van that you were searching on that particular day in May?
Yes, that's correct. The pair of Adidas gloves was actually not contained within the toolbox. It was contained within a smaller lockbox that was recovered under the front seat of the minivan when we did our initial crime scene response search. The smaller lockbox contained a variety of different items, but the Adidas gloves were within that container.
Is that the same lockbox that contained the Walther P22 handgun?
That is correct.
All right. And then you had a known sample from a woman named Michelle Snatella.
That is correct.
Okay. And without beating a dead horse, similar process: swab, dry, cut, tube, and store for later analysis.
Yes, exactly correct.
Okay. Your Honor, I move for the admission of Exhibit 175.
No objection, Your Honor.
Received.
Okay.
Thank you, Mr. Nelson.
Yes, sir. Okay. Handing you what's been marked as People's Exhibit 176. What type of report is this?
People's Exhibit 176 is a copy of the laboratory report that I issued in this case. It is a DNA report. It's identified as record number 18 in the upper right-hand corner. And again, this is a DNA report for one of the subsequent rounds of DNA that I conducted in this case.
All right. So we have added, on top of Ms. Schnatella, we've added a couple other known samples. Is that correct?
Yes, we have.
And that of the new ones, one being Kevin Bloom, right?
That's correct.
And Charlene Ann Willis.
That is correct.
Okay.
Additionally, if I can note real quick.
Yes.
The first two that are listed, the alternate reference from Jessica Herringa and the reference sample from Rebecca Bletch, specific to the time frame on this, those are now DNA profiles based on other examinations in the other cases and the other records. Those are profiles that we had called back to laboratory, and we actually have run these profiles, and I have the more complete and updated, in each of those instances, the full PowerPlex fusion data at this point in the report. So that's the more complete profile. It's not the older, more partial profile as a reference. So this is actually a more complete version for each of those reference samples too.
Okay. And again, this lists the swabs that you analyzed. Is that correct?
That is correct.
Going back again to the interior door handle, the leather straps of the ball gag, correct?
That's correct.
And the swab of the interior, well, I guess there's some new cuttings as well, correct, that you're looking at?
So on this time around, again, starting at the bottom of page 1, these are samples that are now getting DNA analysis for the first time. So we have some new evidence in addition to having some new knowns. So this report gets a little more complicated, but this is the first time we've run the actual interior door handle. Previously I had done the lock, but this is the handle from the van. The leather straps on the ball gag. Previously I had done the ball, and this is the actual swab of the straps being done for the first time. The interior edge of the handcuffs, and that would be the handcuffs that are on the long chain that we talked about. And then other items, the .22 handgun grip and trigger we spoke about earlier, where I actually swabbed the portion that would have been handled on the handgun. That's the first time we see this. And then the last three items in the evidence, the gloves, the Reebok glove exterior, the Mizuno glove exterior swab, and the Adidas glove exterior swab. Each of those items are things we've discussed that are being run here for the first time in DNA analysis.
All right. And then it goes on to the list of things that you have that aren't contained in this report, similar to the other reports that we've talked about.
That's correct. First, the list of things that were previously analyzed and are available for comparison, and then at the bottom of page 2, the other items that still have not seen DNA analysis. They may or may not in the future, but they are not at the time of this report actually analyzed.
All right. So let's go through your analysis here. First, on the interior part of the door, what were you able to determine?
The swab of the passenger side interior door handle did give a partial DNA profile, so it's not dated at all the locations. It did indicate the presence of at least 3 donors. And because it was partial, because it's a complicated mixture, and just the limited amount of actual data that was obtained, no conclusions can be made. It was an inconclusive result.
And I don't. Was this one that was later forwarded on to have the StarMix analysis, or was it it was just a sample that you really can't make any conclusions?
Yeah, this was too partial. At the time that this report was done, at the time of the initial report, we really only wanted to send forward samples for StarMix analysis that we thought had a reasonable chance of giving a good result. This data was so partial, so limited, that there's really no way that a human or a computer is going to be able to make a good comparison. So it was just too insufficient, too inconclusive.
All right, the next swab analyzed was that of the leather straps on the ball gag. What were you able to determine?
Again, the swab of the leather straps in the ball gag were consistent with, it was a partial profile, so it's not at all the locations. It was consistent with a mixture of 2 individuals, and there was a major male contributor that was visible. That was compared against all the reference samples that are available, and the DNA profile from Jeffrey Willis matches the DNA from the major donor on the swab of the leather straps in the ball gag. There's a statistic, it's number 1 on page 9. Probability of selecting another individual at random from the population who also would match that would be 1 in 2.290 trillion in a Caucasian population, 1 in 10.20 trillion in an African American population, and 1 in 7.062 trillion in a Hispanic population.
Okay. And then per your policy, you had to run that analysis for the other knowns you had. Is that correct?
That's correct. I compared against all of the other reference samples that we had at this point in this case, and each of these individuals was excluded as being the major donor. That would be Kevin Bloom, Jessica Heringa, Rebecca Bletch, Madison Nygaard, Charlene Ann Willis, and Michelle Schnatala are all excluded as being possible contributors to that leather straps in the ball gang.
As the major donor.
That's correct, as the major donor. The minor donor from those is insufficient for any kind of a comparison. So yes, each of them are excluded as the major donor. That's correct.
All right. The interior edge of the handcuffs. What was your? And again, these were the handcuffs attached to the chain. Is that correct?
That's correct. The handcuffs that were contained within here that were attached to the chain.
All right.
I did get a DNA profile. It was consisted of being a single source profile from one individual, and it was a male contributor compared against all of the reference samples. And the DNA profile from that swab of the interior edge of the handcuffs matches the DNA profile from Jeffrey Willis. There's a statistic number 2 on page 9. Probability of finding someone else in the population who would also match that profile would be 283.0 sextillion in a Caucasian population, 1 in 220.6 septillion in an African American population, and 1 in 6.135 septillion in a Hispanic population.
All right. And then, again, running against the other knowns you had.
That's correct. The swab of the interior edge of the handcuffs, that single source profile was compared against all of the other reference samples, and each of those individuals was excluded as being a possible donor to the DNA from the interior edge of the handcuffs.
All right. Next was a swab of a syringe needle.
Swab of the syringe needle did give a partial DNA profile. It was from a single source one individual, and it was a male profile. The partial DNA profile from the swab of the loaded syringe needle is actually from an unknown donor, and so I've termed it unknown male donor number 1. At this time, did not know who that is. It is suitable for comparison purposes, so it does have some information that could be usable. And then each of the individuals that I had at this point is excluded as possible donors to that partial DNA profile.
Okay. Next item is the swab of the handgun grip and trigger. What did you find out there?
So the DNA profile obtained from the swab of the .22 handgun grip trigger is consistent with being a single source profile. It is a male profile. The DNA profile matches the DNA profile from Jeffrey Willis. And statistic number 3 on page 9, probability of selecting another individual from the general population who also would match that grip trigger of the handgun would be 1 in 6.124 septillion in a Caucasian population, 1 in 6.369 octillion in an African American population, and 1 in 252.5 septillion in a Hispanic population.
And again, against the other knowns?
That's correct. All of the other reference samples that were submitted at this point were all excluded as possible donors to that grip in 22.
Okay. The swab of a knife, what, if anything, could you find out from that?
So there was a knife that was submitted. I obtained a partial DNA profile. It was a mixture of 2 donors. And because it was such a low amount, it was limited data, no conclusions can be made.
And again, it's of such insufficient quality that it couldn't even go on to any further statistical analysis with the new system. Is that correct?
That's correct. Insufficient quality or potentially quantity, either of those, but there was not enough data that was present for any type of a comparison.
All right. Let's talk about the swab of the Reebok glove, the exterior. What were you able to find there?
Yeah, so again, the Reebok glove exterior was swabbed and run for DNA analysis. The data did indicate that it was a mixture of more than one contributor. It was a mixture of multiple contributors. And because it was a complex mixture, that data was actually forwarded to another scientist so that she could run the StarMix analysis on that to do comparisons.
All right. The swab of the exterior of the Mizuno glove, what were you able to determine?
The swab of the exterior of the Mizuno glove was consistent with being a mixture of 3 individuals. However, this is an instance where one of those individuals was a major contributor, so DNA present in a higher quantity. That major DNA profile matches the DNA profile from Jeffrey Willis. Statistic number 4 on page 9. Be 1 in 473.7 sextillion in a Caucasian population, 1 in 343.9 septillion in an African American population, and 1 in 13.53 septillion in a Hispanic population.
And the other reference samples that you had?
Each of the other reference samples was available at this time were excluded as being that possible major donor. And then specifically of note, the other 2 donors, those minor donors, are low enough on that sample that no type of a meaningful comparison can be made. They're insufficient for conclusive association purposes.
All right. The swab of the exterior of the Adidas gloves.
DNA types were obtained from the swab of the exterior of those Adidas gloves. It was consistent with being a mixture of 2 donors. It did have a major male contributor. A major DNA profile was compared. It matches the DNA profile from Jeffrey Willis. Statistic number 5 on page 9. Probability of selection of an individual from the population who also would match that. It would be 1 in 783.1 septillion in a Caucasian population, 1 in 2.932 non-alien in an African American population, and 1 in 52.66 octillion in a Hispanic population.
All right. And again, run against the other knowns.
Yes, all of the other reference samples that were available at this time were excluded as being that major donor from the swab of the exterior of the Adidas gloves. And then again, the minor donor that's present on those gloves is at a level that's low enough that it's insufficient for any comparison purposes.
Okay. Now, it appears as though the other parts of this report, if I'm looking at this right, were resubmitted. In other words, the ones that we had looked at in record 8, starting at this next one, are being...
Our policy requires that all reference samples be compared to all evidence samples. So the subsequent items in this report are evidence that was run previously, comparisons were made previously, but no comparisons were made to the newer reference samples that were available. So kind of as a matter of housekeeping, from this point out, the items are all of the data from previously getting compared against the new reference samples from Kevin Bloom, Jessica, the updated profile from Jessica Herringa, the updated profile from Rebecca Bletch, the new profile from Charlene Willis, and the new profile from Michelle Santana.
Okay. So were any of those new reference samples found on the swab of the passenger side manual door lock?
So previously that sample was forwarded for evaluation using StarMix, and similarly in this fashion the same analysis is going to apply. That data is still being evaluated using StarMix, but I now made available to that other scientist these 4 new profiles, and she will actually report that out in record number 15.
Okay. I don't necessarily need you to go back over the comparison to the defendant in this case, but when you ran the swab of the red ball gag on the other, or the new knowns, anything else pop out?
Yeah, so looking at the red ball gag, we had a major profile. Kevin Bloom is excluded as being the major donor. Jessica Herring is excluded. Rebecca Bletch is excluded. And Madison Nygaard, Charlene Willis, and Michelle Shantala are all excluded as being that major donor on the ball gag.
All right. Again, the purple dildo.
Yeah, same result. That was previously forwarded for StarMix analysis. So there will be some additional reports. Specifically, the initial one was in 15. Record number 19 will be the new report of those new reference samples compared against that StarMix analysis.
All right. The swab of the silver end of the vibrator.
Yeah, so this is one where we actually get a new association. Remember the swab of the silver end of the vibrator were consistent, the partial DNA types were consistent with a mixture of 2 individuals. There was at least 1 male contributor. A major donor profile was able to be determined originally when I looked at it. It was unknown at that time. Now I can say that Charlene Willis matches the major donor on that swab of the silver end of the vibrator. Because there's an association, there's a statistic, number 7 on page 10. Probability of finding another individual who would match that major DNA profile would be 1 in 9,200,000 in a Caucasian population, 1 in 79,240,000 in an African American population, and 1 in 13,410,000 in a Hispanic population. Excuse me.
Okay. And then obviously run against the new gnomes. Any of those become part of that mixture or a contributor?
Yeah, so again, looking at that major donor from the swab of the silver end of the vibrator, everyone else is excluded. Jeffrey Willis is excluded, Kevin Bloom, Jessica Herringa, Rebecca Bletch, Madison Nygaard, and Michelle Shantala are all excluded as being that major donor.
Your reevaluation of the interior of the leather restraints, any of the new gnomes become any part of the major donor or minor donor of that analysis?
No, all of the new reference samples are excluded as the major donor.
Similarly, the stain on the muzzle end of the 22, any of the new reference samples that you were given part of that mixture as well?
That's correct. All of those individuals are excluded as the major donor on that stain on the muzzle end of the 22.
All right. Again, similarly, your work on the interior underneath the slide, any of the new reference samples part of that profile?
Each of those new reference samples is excluded as the major donor from that stain on the interior of the slide of the 22 handgun.
Okay, the swab of the van cargo floor, again, were you able to do anything with that?
So the swab of the van cargo area floor previously was forwarded for StarMix analysis, and that's referenced in record number 15. This is an instance where in this section I can actually, even without the StarMix analysis, say that Kevin Bloom, Jessica Herringa, Rebecca Bletch, Charlene Willis, and Michelle Shantala are all excluded as possible contributors. It was blatantly obvious from the mixture, given the DNA types that were present, that they were all going to be excluded. So they weren't even compared through that StarMix.
All right. Then we go back to the swab of the waistband of the red panties. What, if anything, were you able to determine?
Yeah, so this is one where previously we had done comparison with Jeffrey Willis, but now having new references available, I'm able to say that, again, that swab of the waistband of the red panties was a mixture of at least 2 individuals. At least one of those individuals is male, but it's not a scenario where we have a major and a minor. There's just a mixture of DNA types. And I could say that both Jeffrey Willis and Michelle Shantala are included as being possible contributors to that mixture of DNA types. Now again, because it's that mixture, the statistic, number 12 on page 11, that statistic is that mixture statistic. So again, it's considering all the different possible pairings and who else in the world could also be included based on all of that data. Be 1 in 930.2 thousand in a Caucasian population, 1 in 18.45 million in an African American population, and 1 in 2.517 million in a Hispanic population. So it would seem that those profiles are rare. However, each of those individuals is included in that mixture of DNA types. All of the other reference samples that were provided are excluded as being possible contributors to that DNA sample.
Okay, and your work on fraction number 1, stain in the pink panty.
Yeah, again, previously, fraction number 1 of the pink panties that stain in the crotch, we had an unidentified donor, and now we are able to say that we can identify, we can see an association. The DNA profile from the stain in the crotch of the pink panties matches the DNA profile from Michelle Shantala. Because there is a match, there's a statistic number 11 on page 11. It's the probability of finding somebody else who also would match that profile. It'd be 1 in 3.621 non-alien in a Caucasian population, 1 in 932.8 non-alien in an African American population, and 1 in 38.02 non-alien in a Hispanic population. Each of those other individuals, each of the other reference samples are excluded as being the possible donor to that fraction 1 of the panties, of the pink panties, the stain in the crotch of the pink panties. So that would include Jeffrey Willis, Kevin Bloom, Jessica Herringa, Rebecca Bletch, Madison Nygaard, and Charlene Willis. Each of them are excluded as being a possible source of that DNA.
And fraction number 2?
Fraction number 2, again, was a mixture of 2 individuals. At least one of those was a male. Similarly, it is a mixture without a major and a minor component, so we're just considering all of the different possible pairings. And both Jeffrey Willis and Michelle Schnatala are included as being possible contributors. There's a statistic number 13. It's a mixture type statistic. It can be 1 in 222.2 thousand in a Caucasian population, 1 in 102.8 thousand in an African American population, and 1 in 317.7 in a thousand in a Hispanic population. Similarly, the other reference samples are all excluded as being in that mixture. Kevin Bloom, Jessica Herringa, Rebecca Bletch, Madison Nygaard, and Charlene Willis. Each of them are excluded as being in that potential mixture, in that mixture of DNA types.
Okay. And so that essentially includes your analysis recorded here in record number 18.
That is correct.
Your Honor, I move for the admission of Exhibit 176, which is record
176?
176.
No objection.
See?
You want to take a break, or you want me to continue? Okay. All right, this is Exhibit 177, which is a different lab number, GR 16-2492, record 22. And specifically, I'm not going to have you go through this entire list here, but this is a serology report, is that correct?
That is correct. People's Exhibit 177 is a copy of my record number 22. On this case, it is a serology formatted report, a serology type of report for screening for potential biological sources.
Okay. In particular, I'm interested in container number 33, container number 34.
Okay.
Container number 33, I believe, was contents of what was in container number 34. But in particular, in 33, what did you do in relationship to the video camera batteries and accessories that were found?
Yeah, so container number 33 was identified to me as being originally within number 34. It had been separated for packaging and inventory. Container number 33 contained a variety of different camera accessories. Imagine camera filters, camera lenses, some different batteries, a variety of different miscellaneous things that could be used on a video camera. Specifically of interest, there was one battery that was located in there, and Find my results. My results on page 4 for item 14O, the video camera batteries and accessories. These results are actually specific to that one battery. The other 20 items were screened for the possible presence of blood. Testing did not indicate the presence on most of them. However, the battery itself did have a hemostix positive result and a phenolphthalein positive result. Again, both of those are chemical tests. A color change indicates the possible presence of blood. So that item was swabbed and forwarded for DNA analysis.
Okay, and then in container number 34, the green camera bag.
Yeah, so container number 34 was a relatively standard green camera bag that would hold a larger format camera. Specifically, my results are listed on page 4 for item 14P. There was some discoloration on the exterior of that bag. As I recall, it was on the bottom of the bag. A little discoloration. I tested it. It was phenolphthalein negative, so it did not indicate the possible presence of blood. Again, then going to that secondary idea, how would an individual have contact with this item? I actually swabbed the shoulder strap of the bag, the portion of the bag that you would grab onto and hold it, swabbed that and forwarded that sample for DNA analysis. And actually, out of all of the items in this report, those are the only 2 items to be forwarded for DNA analysis.
All right. And again, that report is complete. Anything different than when you first submitted it?
Other than some, there's a footer on the bottom for when it was transmitted to the agency. Otherwise, the content of the report does appear as I issued this originally.
Your Honor, I move for the admission of People's Exhibit 177.
No objection, Your Honor.
Received.
Thank you, sir.
Welcome.
All right. Exhibit 178, People's Exhibit 178. This is that same 2492 lab number, and it's record number 25. Is this a serology or DNA report that we're looking at?
People's Exhibit number 178 is a copy of my DNA report. It's record number 25, and it is a complete copy, has my signature on the bottom of page 3.
And does that indicate the DNA analysis you did on the 2 swabs you just previously testified, the one on the camera battery and then the camera bag?
That is correct.
Okay. And obviously, it contains all the known samples you had to run against that.
That is also correct.
What were you able to determine on the camera battery?
So the stain on the camera battery, the resulting DNA, did indicate that it was a partial DNA profile, and it's consistent with a single male contributor. It was a single source profile. That partial DNA profile was compared against all of the reference samples that I had at this point. That partial DNA profile matches the DNA profile from Jeffrey Willis. Again, because there's a match, there's a statistic. It's number 1 on the next page. Probability of selecting someone else who would also match that stain from the camera battery is 1 in 13.12 billion in a Caucasian population, 1 in 11.81 billion in an African American population, and 1 in 35.01 billion in a Hispanic population.
All right. And as it relates to the other knowns that you had?
Yes, each of the other known profiles, reference samples, was excluded as being possible donors. That would be Jessica Herringa, Rebecca Bletch, Madison Nygaard, Kevin Bloom, Michelle Santala, and Charlene Willis. Each of them are excluded as being a possible donor.
Finally, the swab of the camera bag, what were you able to determine off of that?
The data from the swab of the camera bag did indicate that it was a mixture of 2 individuals. There's a major male contributor, and then there's a minor donor, but the minor donor is going to be insufficient for any kind of a comparison. The major donor profile matches the DNA profile from Jeffrey Willis. The statistic is number 2 on the following page. And the probability of finding somebody else who would also match that swab of the camera bag would be 1 in 4.737 quintillion in a Caucasian population, 1 in 236.6 quintillion in an African American population, and 1 in 81.30 quintillion in a Hispanic population.
The other knowns?
Each of the other known reference samples are excluded as being the major donor from that swab of the camera bag.
And then the information you retained from the minor donor, what did you glean anything from that?
Again, that the data from that minor donor is insufficient. It's low enough as far as quality or quantity, so it's insufficient for any kind of a conclusive association.
Your report, I think you've indicated, is intact there, nothing different than the original one you submitted?
Again, other than the footer and actually a small header at the top, a note in the top margin, the content of the report itself is exactly how I produced it.
Your Honor, I move for the admission of People's Exhibit 178.
No objection, Your Honor.
Received.
Okay. Now, this, you were, after the first trial, you were asked to do some additional work, is that correct?
That is correct.
Okay. And that's what we're going to get into right now.
Thank you. You're welcome.
Okay. Handing you what's been marked as People's Exhibit 179. This is record number 30.
Yes.
Now, you had talked initially about taking some swabs way back in May of
Before we get into this, the jurors want to break.
Okay.
So let's take our 10-minute break.
We went fine.
Series of requests for a few more pieces of evidence that was submitted to laboratory in December of last year. I actually analyzed it in January of this year.
Now, some of that evidence, though, you had actually collected back in May of 2016. Is that correct?
That is correct.
And does your report reflect that? And I guess what I'm asking is, does this report indicate which items were actually swabbed by you back in May of 2016?
Yeah, so page 2 of the report, towards the top, container DNA 01-E. That was the envelope that initially contained all of the samples that was collected when I first processed through the evidence. And there's a full inventory listing of each of the items and what they are. In general, the items that appear with an X at the end of the item number, these are items that I've already analyzed. I've already produced DNA results and reported out results previously in the previous reports. The items that end in the letter D are items that are still in the DNA swab form. They have not been processed for DNA analysis, and they still remain in the envelope intact with the swab in the small tube sealed in an envelope. So some of the items have been analyzed, dried down and returned to the agency. Other items have not yet been analyzed at the point that this serology request came through.
And under the conclusions portion of that, is that narrowing down, I think, well, I guess you tell me, is that a narrowing down of the items of the stuff you had already swabbed that were being asked of you to analyze?
That's correct. That conclusion section, the first container, lists the 4 items that actually are still in the swab form. They're in the tubes. And these are the 4 items that were swabbed previously that the request has been made now to actually go ahead and perform the DNA analysis on those 4 items.
Now, below that, the container number 4, these items were also asked of you to be analyzed. Is that correct?
Yes, container number 4 is actually the brown paper bag that contained the original toolbox. And some requests were made for a few specific items, 3 specific items within that toolbox to be swabbed in January, now, currently, to be swabbed and analyzed for DNA analysis. So we have some old items that were already swabbed, and then these 3 items were newly swabbed in the laboratory in January.
Okay. And similar, as we look at L29, again, these were handcuffs, not the handcuffs tied to the chain, but a separate set of handcuffs. Is that correct?
Correct. Originally, L29 I first saw in the laboratory when I was processing through the bulk of the evidence. This set of handcuffs, L29, is the set of handcuffs that were contained within the smaller lockbox that was underneath the seat of the minivan when we recovered it. Subsequent to that, those items all got packaged separately, so they made it into a separate envelope, container number 16, somewhere along the way. But that set of handcuffs was submitted, and the request was made to swab those.
And then the black Adidas gloves.
That's correct. The black Adidas gloves were swabbed. Originally I had swabbed the interior. The black Adidas gloves, originally L37, were recovered originally in that smaller lockbox again that was recovered from underneath the front seat. Originally I swabbed the interior of the other sets of gloves that were in the toolbox itself. I did not swab the interior palm region of the Adidas gloves for whatever reason. So the request was made, let's go ahead and swab the interior of those gloves at this point. So that's when these were swabbed in January of 2018.
Okay. And so essentially this report is a serology report indicating exactly what you just testified to. Is that?
That's correct. It's very procedural, basically just saying a number of other items are being forwarded. No new testing was done as far as testing for blood stains or seminal fluid stains. It's much more procedural. These items are being forwarded for analysis.
All right. Your Honor, I move for the admission of Peoples 179.
Wonder, Your Honor.
You may.
Mr. Harris, look at the last page of that 179 then.
Page 4?
Yes, sir. It says something about record 31 at the top. You see what I'm saying? Am I looking at the thing right now? Yeah, that last page. Right there.
Oh.
Yeah.
Interesting.
Is it interesting? Is it supposed to be there?
So page 4 of exhibit number 179 is not a page that I typically see in the laboratory. I have seen these before because of the way our system works. I have seen these before. Yes, it's this page. This is actually the cover page that I believe our reports are delivered from our system. Forensic Advantage is our internal laboratory computer system, and it is the method by which all of our reports are disseminated to the investigating agencies. In this case, it does list regarding the lab number, GR16-2492, and it does say 31, which to me might be an indication that this actually would be the delivery packet page for the next subsequent report. Okay. But I do not know for sure. This is not necessarily how I view these reports. I actually just have access in the system to the original report that I created. So it is possible this cover page is for the subsequent report.
Check your monitor for a second. That was which should be on Exhibit 30?
Yes, that is correct. That would be my belief. Again, I don't routinely see these pages, but that would be based on my experience with this delivery cover page, the monitor of being number 30 would be probably the correct cover page for this one.
So if you're a betting man, is that just a page that was stapled to that package by inadvertently impression part of that particular report?
Most likely that is probably the best plausible explanation.
Best operator error on that. That's probably my fault.
Your Honor, given that explanation, I don't have further objections to that particular document.
All right, 179 is the
They shouldn't let me around a stapler, I guess. Okay. Next is People's Exhibit 31. Excuse me, People's Exhibit 180. Jeepers. Thank you, sir.
You're welcome.
All right. Handing you what's been marked as People's Exhibit 180. This is Record 31. Is that correct?
Yes, that's correct. People's Exhibit number 180 is a copy of a DNA report that I issued in this case. It is record number 31 as indicated in the upper right-hand corner.
All right. Now, we didn't really cover this in your initial introduction, but you are qualified to run analysis through the StarMix program. Is that correct?
That is correct. I am now qualified. Actually, during the time frame of some of these other reports where I was handing data to another analyst, I actually was undergoing the training process myself. And then I was actually, my full qualification came in December of 2016. And so after that date forward, I no longer had to provide my data to another analyst. I could actually generate my own conclusions using StarMix analysis.
Okay. And this report indicates a carryover, if you will, from the serology report we just went through, indicating your work on the swabs that either had been previously generated or recently generated for this work. Is that correct?
That is correct.
And you also had the knowns as well.
That is correct. And actually, in this case, we specifically received permission to limit the number of knowns to help kind of expedite some of the processing. So very specifically looking for just those 4 known samples for comparison against the evidence. So we did not compare the other reference samples that are involved in this case.
Okay. So this is where we get into, I think, what you were talking about as far as hypothesis and likelihood ratios, and so it's a different set of analysis than what we've gone through over the last couple hours. Is that right?
That's correct.
And just so we set this up, Mr. Hayhurst, in each one of these items, in order to accomplish referencing them to the known samples you had, how many different hypotheses did you have to create, if you will, for each sample?
Yeah, so procedurally, by matter of procedure, we're required to compare each individual separately to a mixture. In each of the instances, the hypotheses are kind of the first step in the process of that comparison. The computer modeling has already been conducted. It has a pretty good idea of the different relative contributors to the mixture, and then I will input a known reference sample. And the first hypothesis is generally that person is included in the mixture. And that would be the hypothesis 1 that's listed here. And then, in general, the second hypothesis is that person's not there. In fact, that mixture is better explained as being just random individuals on the street, a random collection of DNA types. And so again, that StarMix analysis, because it's setting up a likelihood ratio, it's going to weigh each of those possible scenarios and determine which better explains the data. Is it better explained by this person and some other random individual from the population, or is it better explained by being just random individuals from the population? And depending on how the modeling is based, it will produce a likelihood ratio. So that's going to be the number that's represented in that second box of the chart. And I actually have an underline in mine. And then the bottom box is this idea of the verbal scale. Because just giving you a number every time doesn't necessarily help you to determine how much weight on one scenario versus the other. So we also provide a verbal scale. And this verbal scale is set based upon some policies that we have in place, and it's the idea of how strong is that evidence one way or the other. And so there's different levels of verbal scale that are also reported. So it's both a number and then that verbal scale equivalent, so that you can help weigh those 2 scenarios.
All right. The first one is in reference to the swab of the interior of the right and left Reebok glove. Is that correct?
That is correct.
And the first hypothesis was what?
So the modeling was conducted on the interior of the right and left Reebok gloves, and I instructed the computer that this is a mixture of 3 individuals. So the computer went through all the modeling, set its ideas, and then I asked specifically, set it up as hypothesis 1 being that this is best explained by being DNA from Jessica Herringa and 2 unknown, unrelated individuals, or is it better explained by being just 3 unrelated, unknown individuals? Which of these 2 scenarios better explains the data?
And the conclusion that it reached was what?
So based on the DNA typing results from the evidence, it's about at least 1.4 times more likely if it was just 3 random individuals from the population as compared to, or being explained as being originating from Jessica Herringa and 2 unrelated individuals. So there's actually a little larger support for that bottom hypothesis, but at 1.4 times, it's really not much support. So that's where the verbal scale relates that the analysis of this evidence with regards to that reference sample from Jessica Herringa is uninformative. In a sense, it's an inconclusive type of result. It's not strong evidence one way or the other. It's almost to the point where both of them are kind of equally likely type of a scenario.
Okay, then the second set of hypotheses, and this is still related to the Reebok gloves, is that correct?
That's correct. Same piece of evidence being compared this time to a new individual.
All right. And what the hypothesis, the conclusion and verbal scale for this series was what?
Yeah, so on this instance with the comparison of Rebecca Bletch, is it better explained as being a mixture of Rebecca Bletch and 2 unrelated unknown individuals, or is it better explained as being just from 3 random individuals? And in this case, based on the DNA typing results from the evidence, it's at least 310 times more likely if it originated from Rebecca Bletch and 2 unrelated unknown individuals, as compared to being from 3 unrelated unknown individuals from the population. At 310, that is in the area of the scale that we will term moderate support. So this analysis provides moderate support that Rebecca Bletch is a contributor to the DNA typing results from that swab of the interior of the right and left Reebok clubs.
All right. The third set of hypotheses and conclusion and verbal scale.
Yep. So the third set of comparisons, again, the same modeling was done. This time asking it to compare against Jeffrey Willis. Is it more likely that it is Jeffrey Willis and 2 unrelated unknown individuals, or is it better explained as just being DNA from 3 unrelated unknown individuals? And in this case, based on the DNA typing results from the evidence, it is at least 70 quintillion times more likely to be explained as being from DNA from Jeffrey Willis and 2 unrelated unknown individuals, as compared to just 3 random unrelated unknown individuals. So this is an instance where at 70 quintillion, we have very strong support that Jeffrey Willis is a contributor to the DNA typing results from that evidence.
And the fourth hypothesis?
And the fourth comparison that was done, again, is this time comparing Kevin Bloom. Same modeling was done. Is it better explained as being a mixture from Kevin Bloom and 2 unrelated unknown individuals, or is it better explained as just being from 3 unrelated unknown individuals? And in this case, it is at least 49 times more likely that it originated from just 3 unknown random individuals from the population, as compared to being from Kevin Bloom and 2. So this actually, at 49, that's in the area of the region of our classifications that we would call uninformative. There's not a whole lot of support. It's not very strong support one way or the other, so it is termed uninformative.
All right. And that was the analysis of the swab of the Reebok.
That is correct. Specifically, the swab of the interior of the Reebok gloves.
Thank you. Now, the next set of results has to do with the swab of the white rope, and we had one of your counterparts, Sergeant Flowers, kind of show that white rope was contained inside that toolbox. Is that correct?
That is correct.
And essentially, when we say swab of white rope, what does that mean?
So initially, I had screened that evidence for any potential bloodstains and did not see any. It was reported out in one of the previous reports. So initially, did not swab it, go any further with that. The request was made to swab it here in January, and so I swabbed just the length of the rope. I took a swab to try to determine, to see if I could collect any contact DNA from anyone who may have had physical contact with that rope itself.
All right.
So that's the swab that we're talking about here. Right.
All right. The first set of comparisons.
The DNA results did indicate that the swab of the white rope was a mixture of multiple contributors, and it did appear to be a mixture of 4 individuals, so multiple potential DNA donors to this rope. Again, the same set of comparisons is done. The modeling is conducted. Once the computer has a good model for how that data could be explained, then I will ask for the references to be compared. First, I compared the reference sample from Jessica Herringa, and I asked if it was more likely to be explained by being a mixture of DNA from Jessica Herringa and 3 random individuals, unknown individuals, or is it better explained as being just from 4 unrelated unknown individuals. Based on the DNA typing results, it is at least 1.2 trillion times more likely to be explained as being just 4 random individuals, as compared to being Jessica Herringa and 3 unrelated unknown individuals. Excuse me. This does provide very strong support that Jessica Herringa is not a contributor to the DNA typing results. I'm not specifically excluding her as being a donor, but we're starting to get pretty close. It's very strong support that she's not a contributor to that mixture.
All right. Your second comparison?
Second comparison is with respect to Rebecca Bletch. Is it more likely to be explained as being DNA from Rebecca Bletch and 3 unrelated unknown individuals, or is it better explained as being from 4 unrelated unknown individuals? Based on the modeling, it's at least 28,000 times more likely that it originated from 4 unrelated unknown individuals than being explained by being from Rebecca Bletch and 3 unknown individuals. That is very strong support that Rebecca Bletch is not a contributor to the DNA from the white rope.
All right. Similar analysis as before, not excluding her, but
Yeah, we're coming pretty close, but we're not quite to the point where I can sufficiently say she's excluded from this evidence. But it's very strong support. She's not a contributor.
all right. Third comparison.
Third comparison is with respect to Jeffrey Willis. Is the swab of the white rope better explained as being DNA from Jeffrey Willis and 3 unrelated unknown individuals, or is it better explained as being from 4 unrelated unknown individuals? And in this instance, the computer did indicate that based on the DNA typing results, it's 64 million times more likely that it originated from Jeffrey Willis and 3 unrelated unknown individuals than if it originated from 4 unrelated unknown individuals. And at the top of the next page, the verbal scale. This analysis does provide very strong support that Jeffrey Willis is a contributor to that mixture of DNA types from the white rope.
And the final comparison on the white rope.
Yeah, so the final comparison is with respect to Kevin Bloom. Is the data better explained as being from Kevin Bloom and 3 unrelated unknown individuals, or is the DNA more likely to be explained as being from 4 unrelated unknown individuals? And in this instance, it was indicated that it's 280 million times more likely if it originated from 4 unrelated unknown individuals. At 280, that is 280 million. That is very strong support that Kevin Bloom is not a contributor to the DNA typing results from that white rope.
All right. Next item was the swab of the chain, and this is not the chain with the handcuffs, right? There was a separate section of chain.
I can check my notes real quick.
Yeah, please.
That is correct. Item L13 was just a length of chain. In addition to the chain with the handcuffs, there is a separate section of chain that was contained within that original toolbox. And in this instance, that request was made to swab that chain for any, again, possible contact DNA that was left with respect to individuals who might come in contact with it.
All right, and go ahead with your first comparison, please.
So again, data was obtained, indicated a mixture. I determined that that mixture was 3 individuals. It was a mixture of 3 different donors. And the first comparison with respect to a reference sample was against the reference sample from Jessica Herringa. And was that mixture better explained as being DNA from Jessica Herringa and 2 unrelated unknown individuals? Or is it more likely that that DNA is representative of 3 unrelated unknown individuals? And in this instance, based on the data, the DNA typing results, it's at least 400 times more likely that it originated from 3 unrelated unknown individuals than being a mixture of DNA from Jessica Herringa and 2. At 400 times, that provides moderate support that Jessica Herringa is not a contributor to the DNA typing results from that swab of the chain.
All right. The second comparison.
Okay, so the second set of questions, again, is with respect to Rebecca Bletch. Is the DNA better explained as being from Rebecca Bletch and 2 unrelated unknown individuals, or is it more likely to be explained as being from 3 unrelated unknown individuals? Based on the data, based on the DNA typing results for the swab of the chain, it is at least 160 times more likely that it originated from 3 unrelated unknown individuals than if it was from Rebecca Bletch and 2 unrelated unknown individuals. That is moderate support, provides moderate support that Rebecca Bletch is not a contributor to the swab of the chain.
Okay, the third comparison.
Excuse me, third comparison, again, the swab of the chain compared against the DNA profile from Jeffrey Willis. Is it more likely that the DNA is a combination of DNA from Jeffrey Willis and 2 unrelated unknown individuals, or is it more likely that the DNA is from 3 unrelated unknown individuals? And based on the DNA typing results, it is 820 sextillion times more likely to be explained as being DNA from Jeffrey Willis and 2, as compared to being DNA from 3 unrelated unknown individuals. So this provides very strong support that Jeffrey Willis is a contributor to the DNA typing results from the swab of the chain.
And the last comparison.
The last comparison is with respect to Kevin Bloom. Is this evidence the swab of the chain better explained as being from Kevin Bloom and 2 individuals, or is it better explained as being from 3 unrelated unknown individuals? And it is 3.4 thousand times more likely that it's explained as being from 3 unrelated unknown individuals than if it was Kevin Bloom and 2. This provides strong support that Kevin Bloom is not a contributor to the DNA typing results obtained.
All right. Next, this swab of the ball on the J hook. Again, I think this was one that you had done back in May of 2016. Is that correct?
That is correct. The ball end of the J hook was swabbed in May of 2016.
Okay. And your first comparison?
Specific to the ball of the J hook, a DNA profile was not obtained for whatever reason, whether it's a quantity issue or a quality issue. There was not DNA that was sufficient for DNA typing. No profile was obtained, so no comparisons were done.
Then, you stuck with the J hook, but you swabbed at different locations. Is that correct?
That's correct. The other end, the eyelet end at the top of the J hook, was swabbed here recently in January of 2018. That sample is the next one that's represented here.
Okay. Your first comparison, please.
That did indicate the swab of the handle and eyelet of the J hook did indicate a mixture of 2 individuals. So the computer did its modeling and got its understanding of who the donors are, or what the DNA types from the donors are. Specifically, when comparing against Jessica Herringa, is it better explained as being DNA from Jessica Herringa and 1 unrelated unknown individual, or is it more likely that it is from 2 unrelated unknown individuals? It is 180 times more likely to be from 2 unrelated unknown random individuals than being Jessica and another. This provides very strong support that Jessica Herringa is not a contributor to the DNA typing results from that eyelet, the hook end of the J hook.
And it looks like you're able to, you found an exclusion as well.
Yeah, so this is, the next instance is an example of an exclusion. The DNA typing results from Rebecca Bletch, the DNA profile from Rebecca Bletch, was significantly different enough, and it was absent, the DNA types were absent, that the software actually was able to exclude her as being a possible contributor to that, the eye of the handle of the J hook.
All right. The next comparison.
Next comparison was against the DNA profile from Jeffrey Willis. Again, is it more likely to be explained as being DNA from Jeffrey Willis and an unrelated unknown individual, or is it more likely that it is from 2 unrelated unknown individuals? And in this case, it was 990 sextillion times more likely that it is from Jeffrey Willis and 1 unrelated unknown individual than if it were from 2 unrelated individuals. This analysis provides very strong support that Jeffrey Willis is a contributor to the DNA typing results.
And you were able to determine another exclusion in this one.
Yes, again, the next point is with respect to Kevin Bloom. The DNA types from Kevin Bloom are different enough, are significantly, they are different from the DNA profile from that swab of the J hook eyelet, and so he is actually excluded as being a possible contributor to the DNA typing results from that J hook.
All right. The next results were from the swab of the interior of the black Mizuno gloves. Is that correct?
That is correct.
And your first comparison?
Again, the DNA from the black Mizuno glove did give a mixture. It was a DNA profile that was a mixture. It did appear to be 3 individuals. So the computer processed its modeling, and then I asked it to compare against the DNA profile from Jessica Herringa. And I asked if it was more likely to be explained by being Jessica Herringa and 2 unrelated unknown individuals, or is it more likely that it is from 3 unrelated unknown individuals? And in this case, it is at least 55 billion times more likely that it was from 3 unrelated unknown individuals. So this analysis, it provides very strong support that Jessica Herringa is not a contributor to the DNA from the interior of that black Mizuno glove.
Okay, your second comparison, please.
Second comparison is with respect to Rebecca Bletch. DNA profile explained as being from Rebecca Bletch and 2 unrelated unknown individuals, or is it better explained as being just 3 unrelated unknown individuals? And in this case, it was 11,000 times more likely that it's just from 3 unrelated unknown individuals. This provides very strong support that Rebecca Bletch is not a contributor to the DNA swab from the interior of the black Mizuno glove, singular, excuse me.
All right, your third comparison, please.
The next comparison is with respect to Jeffrey Willis. Is it more likely that it is explained as being from Jeffrey Willis and 2 unrelated unknown individuals, or is it more likely to be from 3 unrelated unknown individuals? And in this case, it's 66 sextillion times more likely to be explained as being from Jeffrey Willis and 2 unrelated unknown individuals. And this provides very strong support that Jeffrey Willis is a contributor to the DNA typing results from the interior of the black Mizuno glove.
All right, and the final comparison?
The last one was with respect to Kevin Bloom. Is it more likely that it is explained by being from Kevin Bloom and 2 unrelated unknown individuals? Or is it more likely that it is from 3 unrelated unknown individuals? And in this case, it's 25,000 times more likely that it's from 3 unrelated unknown individuals. Provides very strong support that Kevin Bloom is not a contributor to the DNA typing results from that black Mizuno glove.
Okay, then the next comparison is in regards to the swab of the handcuffs, the interior, excuse me, of the handcuffs. And again, these were handcuffs found not in the toolbox, but in a separate box underneath the driver's seat.
That's correct. Item L29, the handcuffs were the one from the smaller lockbox underneath the seat. DNA profile did, it was a mixture of 2 contributors. And so then I compared it against the reference samples that we had.
You were able to, there was an exclusion, is that correct?
Yeah, so computer built its models to get its understanding of who the contributors, what the contributors' DNA profiles might be. I asked it to compare against the DNA profile from Jessica Herringa, and it indicated that she is excluded as a possible contributor to the DNA obtained from that swab of the handcuffs.
Excuse me, your second comparison?
Second comparison was with respect to Rebecca Bletch. Asked it to model whether it's more likely that it was Rebecca Bletch and 1 unrelated unknown individual, or is it more likely that it's just 2 unrelated unknown individuals. And in this instance, it is at least 210 billion times more likely to be explained as being from Rebecca Bletch and 1 unrelated unknown individual than if it were from 2 unrelated unknown individuals. And this analysis provides very strong support that Rebecca Bletch is a contributor to the DNA typing results from that swab of the handcuffs interior.
The next comparison, please.
The next one is specific to Jeffrey Willis. Is it more likely to be explained as being from Jeffrey Willis and 1 unrelated unknown individual? Or is it more likely to be explained as being from 2 unrelated unknown individuals? Based on the DNA typing results, it is at least 250 million times more likely to be explained by being from Jeffrey Willis and 1 unrelated unknown individual. And this provides very strong support that Jeffrey Willis is a contributor to the DNA typing results from that swab of the handcuffs.
And then you were able to form another exclusion in that, is that correct?
That is correct. When compared against the DNA profile from Kevin Bloom, the computer system did indicate that he is excluded as being a possible contributor to the DNA that was obtained from the swab of the handcuffs.
Okay. Let you take a drink.
Sorry.
You're fine. Next comparison involved the swab of the interior of the Adidas glove.
That's correct.
And again, these Adidas gloves were inside that smaller safe underneath the driver's seat. Is that correct? if I'm getting that correct?
Let me
Or
double
they
check
found
that.
somewhere else.
Yes, that is correct. The black Adidas gloves were within the smaller lockbox, not within the larger toolbox. The black Adidas gloves, item L37, were within the smaller lockbox.
All right. You were able to, it looks like, come to some exclusions right off the bat. Is that correct?
Yeah, so the DNA profile actually indicated that it was from 1 individual. So this is an example of an instance where I could revert back and do the random match type statistic. but in order to maintain continuity with this report, I elected to do this comparison using the likelihood ratio that is built into StarMix. So it is an option to run single source profiles here, but the DNA that I obtained was from one individual. I was able to exclude Jessica Herringa, Rebecca Bletch, and Kevin Bloom as being a possible donor to that DNA.
All right. The one comparison you did then is what?
Yeah, so the one comparison that I did do was, after it built its model of a single source donor, I asked it to compare it against the DNA profile from Jeffrey Willis. And is it more likely that this DNA, this single source profile, is from Jeffrey Willis, or is it more likely that this profile is from some random individual in the population, unrelated and unknown? And it is at least 20 quintillion times more likely that it's from Jeffrey Willis than if it was from a random person from population. This analysis provides very strong support that he's a contributor to the DNA results that I've found.
Okay, and I think last but not least, the last analysis was on a stain taken from a tissue box. Is that correct?
That is correct.
And this tissue box, was this also located in the van?
That's correct. In the rear of the van, we recovered item L40, which was a standard size tissue box. It was in the rear of the van, and on the top of the box there was a small area of staining that did indicate the presence of blood, so it was forwarded for DNA analysis, and that's what's analyzed here. I did obtain a single source DNA profile from one individual. I was able to compare that and sufficiently exclude Jessica Herringa, Rebecca Bletch, and Kevin Bloom as being a source of that DNA. And then I compared it against the DNA profile from Jeffrey Willis. The DNA, hypothesis one would be that the DNA is from Jeffrey Willis, or is it more likely to be explained that the DNA is from a random, unrelated, unknown individual? And based on the results that I obtained, it is 29 septillion times more likely to be explained as being from Jeffrey Willis as compared to a random person. And this does provide very strong support that he's a contributor to the DNA typing results from that stain on the tissue box.
And just, I know you kind of briefly talked about that at the end, but just above your signature is the chart that you've been referencing throughout this entire piece of testimony. Is that correct?
That's correct. This is the chart where we will translate that likelihood ratio number to the verbal scale equivalent. Again, this is our effort of trying to give you the best indication of how strong this evidence is. If the two hypotheses are equal, then the number is actually 1, and that's where anything around the number 1 is basically either one of those things is possible. As we get further away from the number 1, then we start to see support for one or the other hypotheses. And so the further away we get, the larger the number, that's the greater support that we see here. So these are the ranges that we start to see, or that we reference in our reports. Anything greater than 10,000, it's 10,000 times more likely that we have one scenario than the other scenario. Well, that's where we start to term it very strong support.
And that report is, I guess, is a copy of the original that's bearing your signature. Is that correct?
Yes, this report has 11 pages. It does bear my signature at the end. There is a small footer that's present at the bottom, but again, I believe that's a part of the delivery system that our computer software has. Otherwise, this report does appear intact.
All right. Your Honor, I move for the admission of People's Exhibit 180.
No objection.
Received.
All right, there's just one more, I promise you, just one more that I have for you. I just have to find it. All right. Well, I apologize. I guess I didn't put it. This is going to be back referencing GR 132559. Record number 15. All right. Handing you what's been marked as People's Exhibit 181. And I apologize to the jurors. I guess I didn't put that in my little PowerPoint. But start of your testimony, you had done some work on that Walther battery cover.
That's correct.
Now, I'm assuming that when you did that initial work, that was under the old system that you talked about.
That's correct. Originally, that DNA analysis was conducted under what we call PowerPlex 16 Hot Start. It's the previous generation of DNA technology that looks at 16 locations. Subsequent to that, we've moved to PowerPlex Fusion, which looks at 24 locations, and it's quite a bit more sensitive.
Okay. Now, did you re-look at that analysis of the battery cover at some point in time?
Yes. In the summer of 2016, in conversation with the detectives, and I think some of it started with wanting to get an updated reference profile for Jessica Herringa, we asked that that evidence be resubmitted to the laboratory. But again, in looking back at my original data and some of the quantitation data that I saw for that battery cover, led me to believe that there may be some benefit to redoing that sample, reanalyzing that sample with our updated chemistry technology. Because the PowerPlex Fusion is quite a bit more sensitive than the previous generation of technology, I didn't wonder if we wouldn't get more data, more of a usable type of a DNA profile to do some comparisons. So that was one of the items of evidence that I requested be submitted to the laboratory so that we could kind of take another shot at it, again, using that updated, more sensitive technology and see if we could glean any more information. And
did you run that through that technology?
Yes, I did.
And what, if anything, did you see?
So that's the results are reported here in my record number 15. It's People's Exhibit 181. Again, previously I had determined that it was a mixture of multiple contributors, but previously the DNA was too low for any comparisons. This time around, using the newer technology, I did obtain, again, a mixture of multiple contributors, and I did forward the data for StarMix analysis. It was a mixture. It was a complex mixture. It was not a clear-cut major-minor type of a situation. So I elected to forward that data to another scientist to do that StarMix analysis.
And was there any, for example, based on what you had in front of you, could you see either female or male parts of that mixture? What could you see?
Yeah, so I could tell that it was a mixture of multiple contributors. There was some indication that there was at least a male donor present. And at the time, because at this time frame we only had 2 scientists in the Grand Rapids laboratory who were trained in the StarMix analysis, we were trying to be very careful and not overwhelming them with a lot of comparisons that maybe would not yield fruit. And so by doing some comparisons, just roughly between some of the reference samples that I had and the mixture of DNA that I had present, I believe that there was sufficient DNA that there might be some value to doing this comparison. It was not likely to be a waste of time necessarily. So kind of looking at the data itself, yes, it was a mixture. Yes, I think that there are some conclusions that could be drawn, and then I forwarded it and requested that it be compared against the different individuals. The results of that are actually reported in a subsequent report to this.
Yeah, in fact, I believe one of your counterparts, Michelle Schmidt, actually ran that analysis.
That is correct.
Okay. Okay. And essentially, essentially your report is saying, looks like maybe I'm going to forward it on to have somebody do the StarMix analysis, essentially?
That's correct.
Your Honor, I move for the admission of Exhibit 181.
Okay. Proceed.
Well, I'll leave it at that. I think, sir, that I have no further questions.
You, Mr. Johnson?
Hey, sir.
Good afternoon.
Good afternoon.
Good afternoon.
Good afternoon, sir. Thank you for your patience with us this afternoon.
Thank you.
Are you okay? You need water or anything?
Doing okay.
Okay. All right. Now, as I sat there listening, understanding I'm a lawyer and I'm certainly not a statistical nerd. It sounded to me like there were two ways that you were analyzing these materials with exhibits, basically 1 through 29, and then you went to the modeling with the exhibit. I think it was 30. Do you understand what I'm getting to?
I think so. Some of the comparisons were conducted kind of asking and answering one set of questions using one statistical tool.
Okay.
And some of the comparisons were using a different asking and answering with a different statistical tool.
Okay, thank you. That's much better said.
Fair enough.
For the first statistical tool, the ones 1 through 30 or 29, whatever the number happened to be, I listened to the as well as I could understand it, and you received on one occasion a blood sample that you were able to determine was the sole contributor was Ms. Herringa. Do you recall that?
Yes, I recall there was a blood stain that gave a female DNA profile. When it was compared to the DNA profile from Jessica Herringa, there was a match.
Okay, good, a match. All right, and do you have any idea how old that stain was before it was gathered and sent to you?
No, I do not.
How long it was on the ground or anything like that?
No, I do not.
Okay, and that one you found matched Ms. Herringa, is that correct?
That is correct.
But the remainder of the materials, up to 30, there were no Herringa matches, were there?
Based on my recollection, no.
So there were, and you were the one who gathered some of this material during a search of my client's vehicle?
That's correct.
Okay, so you went through his vehicle, and you didn't find any matches in his vehicle?
Based on the DNA samples that I tested from the vehicle, no associations were made to Jessica Herringa.
And that would include the passenger seat, front passenger seat?
That's correct.
That would include the cargo area?
The swab from the cargo area, no, she was excluded as being a donor.
That would include the toolbox itself, the black toolbox itself?
The actual toolbox itself was not analyzed for DNA, but several of the items within were analyzed.
Any Herringa matches in the items within?
She was excluded from each of the items that were tested within that toolbox.
Okay. All right. Now, then 34, you used a different statistical model, correct? You're talking about comparisons between whether it's this person versus and 2 others, for example, and 3 complete strangers, that sort of thing, correct?
So in People's Exhibit 180, which was my record number 31, that is the first instance where I am using the StarMix to determine that likelihood ratio.
Okay. That's why it's different. Okay, I missed that part. Thank you. All right. So, given that secondary system, the StarMix system, starting with 31, did you find any comparisons where it was more likely Ms. Herringa and another person, or Ms. Herringa, or a group of people who are not Ms. Herringa?
No, in each of those instances, either she is excluded or there is support against her being included in that mixture, or in one instance it was just uninformative, so it was inconclusive. But there are no positive types of association with Ms. Herringa.
Okay, thank you. That chart, good. Thank you, Ms. Nielsen. That chart's still on the screen behind you. Is it the likelihood ratio chart? I guess quantitative equivalent.
Yeah,
chart?
that's our verbal equivalent for when we discuss likelihood ratios.
And I expect that your colleague Michelle Schmidt will be called in this case. And does she use that same chart in her work?
That is correct. As a part of policy, we are required to put this section of text. The 2 paragraphs in that chart have to go in every report that uses this technology.
Okay, thank you. I've got a few more questions, but they're scattered around here, so forgive me. You were the one who seized the toolbox, the black toolbox, correct?
I don't know that I'm necessarily the one who found it where it was recovered, but after I was there when it happened, and after we recovered and removed it I was the one who snapped the lock on
Can
it.
you tell me why it was cut open as opposed to just using the key?
We did not have a key that we knew to be the matching key for that specific lock at that time.
Did you retrieve this toolbox from my client's vehicle?
I was there when it was, I don't remember if I physically lifted it out of the storage compartment, but I was present when it was recovered.
So the vehicle was open at the time?
That is correct.
But the keys weren't in the vehicle, is that correct? If you recall?
I really don't recall. If we had, frequently we do have keys available, sometimes we just have the unlocked vehicle. I do not recall in this instance.
Was the lock itself that they held the toolbox closed, was that preserved for your purposes?
The lock?
Yeah, that you cut.
Yeah, so the lock, after it was cut, I believe we packaged the lock perhaps within the toolbox.
Okay.
I don't remember exactly where it's at this point.
Okay. Way back in the beginning when you started this ordeal, you talked about the nature of DNA and how obviously blood is a great carrier of DNA, but you can leave DNA by touching a tabletop or something along those lines. And my question to you is this: when you found those tools in the toolbox, were they separated by any mechanism, by plastic bags or restraining walls or compartments, or were they just all jumbled in together?
Primarily, all of the items that were contained within the toolbox were found in approximately the same condition they are now.
Okay.
They were all commingled. There was one smaller case within the toolbox that contained a handful of ammunition and a pair of rubber gloves, or nitrile gloves. They were contained within that box and protected from the other items. But other than that, each of the items in that toolbox was, in a sense, commingling with the other items.
And once you finished removing them, swabbing them to get your sampling, what happened to those items?
Yeah, because the items were originally recovered in this fashion, in this case I elected to leave the items in this fashion. So as the items were processed in the laboratory, they all went back into the toolbox as it appears now, and have remained in that condition since then.
And then at some point, at least some of the tools were retested?
That's correct.
Okay, so they were removed from the toolbox again?
That's correct.
Okay, here's my question. Having established that, what's to prevent one item with DNA on it from rubbing up against another item with DNA on it and transferring the DNA from one object to another?
Yeah, that's a very real possibility. And that's typically something where we will, as we're processing and collecting evidence, evaluate whether each of those items should get sealed separately in a container or not. In this instance, as I was on scene and the other analysts that I was with, we determined that the items had already all been together in the toolbox when we recovered them. So to leave them in that same condition would not have deleterious effects. Any transfer that may have already happened may have already happened. You know, so we made no effort at that time to separate each of those items.
So you were cognizant that transfer may have occurred with these items?
Yes, that's very real.
Okay.
Yes.
I noticed when you do your DNA work and your serology work, you use separate reports. Is that your personal standard of operation?
Typically, we have always done them as separate reports. There actually is one of the exhibits that was a combined biology and DNA report where we basically handled it all as one. That's kind of the way of the future across the state.
Okay.
But typically, for all of the other records in this case, you are going to see a serology report that is separate from a DNA analysis type report.
All right. Sir, did you, when you, I assume you take time off?
I do occasionally.
Occasionally. And when you do that, what happens to your work? What happens to the samples that you are given custody of, control over, or you're responsible for?
Yeah, so any of the cases that I am working that have been assigned to me are in my custody while I am processing them, and at times when I am not processing them, they are stored in a secured location. We have several refrigerated or freezer type vaults that we can use that are secure locations, and we have a chain of custody that tracks as it goes in and out of some of those locations. But yeah, while I am gone, for cases that have been assigned to me and they are my responsibility, they will get stored, and other analysts do not necessarily have access to that evidence.
That was my. You said they don't necessarily have access to. On what circumstances does somebody else? Can somebody else go in and work with your materials, the materials you're responsible for?
So let me maybe clarify that. Maybe that's not the best word. It's stored in a secured location. Other DNA scientists at my laboratory could potentially have access to it. They have the same key card that I have to get into that access location. So it's not that they don't necessarily have access. They just necessarily don't have reason to go in the hundreds of samples that I may have at any given time to go in and have access to my sample or physically have contact with my samples. I guess in theory they could access them if they are a member of our DNA unit, but there are codes and such in place, key cards that are limited to different individuals in the Grand Rapids laboratory. I don't have access to the drug vault, and they don't have access to my vault.
Okay, and I would don't assume, ask. I got you right here. People in your unit have access to the secure areas where you have access to.
That's correct.
Others do not. And other law enforcement officials or any managers. Does anybody else besides your group have access to that secure area?
Yeah. Any of the DNA scientists in our laboratory will have access. The DNA unit supervisors would have access. The lab director would have access if he would know where to necessarily even look. And I believe that should limit the number of people who have access. Obviously, outside law enforcement do not have access to our vaults or any of our secure laboratory examination areas.
All right. Okay. Thank you. I have a couple of technical questions. I'm trying to read these reports, and now that I got you, I'm going to pick your brain a little bit. You mentioned that the systems that are used to do the DNA analysis aren't necessarily conflicting. In other words, if you use the PowerPlex, you can use another. Can you use a StarMix without getting information that's incorrect?
Okay, so not necessarily. The two are not compatible. So PowerPlex 16 Hot Start was that previous generation of kit chemistry that we would use. We have updated that to the next generation of PowerPlex Fusion. It gives us a more complete picture of the data. The StarMix analysis that we use to help model the DNA actually was specifically designed and implemented for use only with Fusion. So I can't pull out a cold case from 10 or 15 years ago and analyze it with that StarMix statistical software. If I want to look at old mixtures, I actually have to rerun them in the PowerPlex Fusion technology, so bring it up to the current standard, and then I can apply that data through StarMix, which is actually part of the reason that battery cover was reanalyzed. Wanted to bring it up in the newer technology in the hopes that I might have more information that could be used in that computer modeling program.
Analyzing it once doesn't use it up. In other words, it's not consumed. It's still available for reanalyzation.
It did certainly use a little bit of the analysis. Typically, we'll bring our DNA extracts up into a 40 microliter volume. I used some portion of that, but there was still some DNA left that was dried, returned. When it came back to the laboratory, there was still enough DNA that I could do some retesting. Potentially, there's more that I could do retesting if we have a next generation of technology that comes on board. So there's still some amount of DNA that is left behind.
Mr. Hazer, what is a reference sample?
So reference samples are otherwise known as known type samples. Reference samples are submitted to the laboratory so that we have a known sample for comparison from different individuals. Typically we use buccal swabs, but we have other forms too.
All right, thank you. May I approach you on it? I'd like to show you a couple documents that I'm working with here. Do you know what those documents are? Have you seen those before?
So these documents are 4 printed pages that do appear similar to some of the printouts from our document or our computer system that we use within the laboratory for tracking our evidence.
Okay.
I don't know how complete they are or at what point they were captured as evidence for case may grow. We may have more or less than what we see here, but they do appear somewhat consistent with what I've seen.
Do you know who inputs that stuff, who inputs that information in those documents?
So initially, as the containers, the outer containers are brought into the laboratory, they are received into the system by our front desk, by our evidence technician, our secretaries, and they leave them sealed. They don't go poking through the container. So the outer container would have been labeled originally and broken down in the system. And then as I'm doing the analysis, I will open that outer container and inventory each of the items within. And so some of these items appear to be consistent with the stuff that's done by the secretaries and our evidence technician, and some of these would appear more consistent with items that I have broken down in the laboratory as I'm doing my analysis.
Thank you, Mr. Hayes, sir. Thank you very much. I don't have any further questions.
Okay, thank you. You may stand down.
Can I just ask him one follow-up, Judge?
Okay.
Please. Just on the issue of transfer, in the particular case, in reference to the toolbox, was there a large dried blood stain in that box?
There was not. As we initially inventoried the item, opened it and inventoried it at the scene and listed all of the items, there was not any blatantly obvious large stain that was present. That might have changed some of the determination in how I would preserve and store that evidence, but we did not see any obvious indications of visual red-brown staining on any of those items.
And I'll just expand it to other fluids. I mean, in other words, was there some source of fluid that would be transferring all this DNA all over the place if this stuff was together?
Not that was visible using our standard lighting that we had at the scene. There was no obvious or blatant source of DNA that was within that box.
And certainly, sir, in your experience, whether or not there's a transfer, would you find that the quantity to be a lot less, as opposed to if it came from a source like a stain, like a blood stain, saliva stain, semen stain?
Yeah, again, this would be an instance where the quantity would be important. If we had something that had a large quantity of stain being present, I might expect that we would see some transfer of that DNA. But if we have something where there's really a minimal quantity to begin with, any transfer is likewise to be pretty minimal. I'm not going to expect to see a large transfer from something that had very little to begin with at the source.
All right. Thank you. That's all I have, Judge.
No, sir. Thank you very much.
Thank you, Mr. Havers.
Thank you.
Next witness, please.
Yes.
Mr. Hayes.
People of the state of Michigan call Michelle Schmidt to the stand.
Please raise your right hand. Do you swear that the testimony you're about to give will be the truth? Please raise your right hand. Do you swear that the testimony you're about to give will be the truth, the whole truth, and nothing but the truth, so help you God?
I do.
Please have a seat, state your name. Spell your name, sure, I'll bring one in.
Thank you. Michelle Schmidt, Michelle, Schmidt, Schmitt.
Thank you, Charles.
Yep. Okay, give me 1 second here. Close all this stuff down. Okay. Okay. Mr. Johnson, I'm not going to go through qualifications unless you want me to.
I do not require you to go through qualifications.
All right.
Thank you.
Given her prior testimony, I'd move to admit Ms. Schmidt as an expert in DNA.
Okay, this witness will be
Analysis.
allowed to be examined as expert in DNA analysis.
Okay. I'm going to start my questioning of you at, I only have a couple of records here that I want to go through with you, Ms. Schmidt. First is, for your reference, is GR 16-2492, record 15.
Figure 182.
This is Exhibit 182. Oh, I'm sorry. Okay. Handing you what's been marked as People's Exhibit 182. Give that to you. Is that a report that you authored, ma'am?
Yes, it is.
Okay. And in particular, you are one of the analysts at the crime lab that is certified in using the StarMix program. Is that correct?
Yes, I have been trained and I am proficient in using the StarMix program to calculate likelihood ratios in mixtures and in single source samples.
And in this particular, you had some swabs that you were running through that program. Is that correct?
Yes, I had some electronic data that David Hayhurst passed on to me to perform likelihood ratio calculations on.
So I want to get to the comparisons. The first comparison had to do with a swab from a 22 caliber round. Is that correct?
Yes, the first item that was tested on this record was a swab of a .22 caliber round.
And were you able to make any comparisons based off of the computer modeling or data?
No, when I tried to model the data, it did not meet quality criteria within our laboratory. Therefore, no known samples were compared to that DNA sample from the swab of the .22 caliber round.
Okay. And the next level of comparison had to do with a swab of the passenger side manual door lock. Is that correct?
That is correct.
And what were you able to? What comparison were you able to make?
And for this sample, it was a mixture of DNA, and a mixture of DNA means that the source arises from more than one individual. And in this case, it was a mixture from two individuals, from two people. And I separately compared the known DNA samples I had at the time. I first compared the DNA sample of Jeffrey Willis to that mixture from two people on the swab of the passenger side manual door lock. And to do that, I set up two scenarios to see which one of the two scenarios better explains the DNA that was detected on that sample. So the two scenarios I compared, I set up like a fraction, and on the top half of the fraction, looking at the DNA profile of Jeffrey Willis, I wanted to see is the DNA obtained from the swab of the passenger side manual door lock. Is that DNA from Jeffrey Willis and one unknown unrelated person? Or is that DNA better described as originating from two unknown unrelated people, neither of whom are Jeffrey Willis? And based on the DNA results for that sample, it was at least 790 million times more likely that it originated from Jeffrey Willis and one unknown, unrelated individual compared to if it originated from two unknown, unrelated people. And based on that analysis and that calculated statistic, there is very strong support that Jeffrey Willis is a contributor to the DNA results obtained from the swab of the passenger side manual door lock.
And there is an exclusion here too, is that correct?
That is correct.
Who was excluded?
At this time, I was able to exclude Madison Nygaard as a possible contributor to the DNA obtained from the swab of the passenger side manual door lock.
Okay. And the other comparisons that you made, or excuse me, the next comparison that you made was based off of the swab of the end of a dildo vibrator, is that correct?
Yes, it is.
Okay. And what comparison were you able to make here?
This DNA sample from the swab of end of dildo/vibrator was also a mixture of DNA, and that mixture of DNA was from two different people. So again, I separately compared the two knowns I had at the time that this report was written. I first compared the DNA profile of Jeffrey Willis to that mixture of DNA obtained from the swab of end of dildo vibrator. So I set those two scenarios up to see if given the evidence, if Jeffrey Willis was a possible contributor, or if given the evidence, Jeffrey Willis was not a possible contributor. So those two scenarios that I compared were the first one, the DNA from the swab of end of dildo vibrator is from Jeffrey Willis and one unknown unrelated person, or is that DNA from the swab of end of dildo vibrator better is explained as originating from two unknown unrelated people. And based on the DNA results for the swab of end of dildo vibrator, it was at least 79 billion times more likely that it originated from Jeffrey Willis and one unknown unrelated individual compared to if it originated from two unknown unrelated people. And based on that analysis and calculated statistic, there is very strong support that Jeffrey Willis is a contributor to the DNA results from the swab of the end of the dildo vibrator.
And again, were you able to make any exclusions?
At that time, I was able to exclude, I had the DNA profile of Madison Nygaard, and she was excluded as a possible contributor to that DNA sample from the swab of end of dildo vibrator.
All right. Your next comparison involved a swab of the cargo van area floor. Is that correct?
That is correct.
Okay. And what, if anything, were you able to conclude from that?
That sample, that DNA sample, the swab of van cargo area floor, was a mixture of DNA that originated from three people. So it was a mixture of three people's DNA present in that sample. The two scenarios that I compared when looking at the DNA profile of Jeffrey Willis is the DNA obtained from the swab of van cargo area floor, better is explained as originating from Jeffrey Willis and two unknown unrelated people, or is that DNA obtained from the swab of the van of cargo area floor, is that better is explained as originating from three unknown unrelated people, none of whom are Jeffrey Willis. And based on the DNA results for the swab of van cargo area floor, it is at least 14 million times more likely that it originated from Jeffrey Willis and two unknown unrelated people compared to if it originated from three unknown unrelated people. And based on that analysis and calculated statistic, there is very strong support that Jeffrey Willis is a contributor to the DNA results from the swab of van cargo area floor.
Okay. And again, were you able to make exclusions?
Yes, at that time I was able to exclude Madison Nygaard as a possible contributor to the DNA obtained from the swab of the van cargo area floor.
All right. Now, in your remarks section here, you talk about asking for, or basically making remarks on comparisons to two other individuals, Rebecca Bletch and Jessica Harrington. Is that correct?
That's correct.
And essentially asking for are you asking for those samples to be resubmitted to make comparisons with those as well?
Yes, at the time those were originally run, we were using a different amplification system. And to make comparisons, they all needed to be run in the same amplification system. And an amplification system is where we make millions of copies of the areas that we test for, where we can then view it in graph form and make comparisons. So at the time that all these comparisons were made, we were looking at 24 locations where we had done testing. And previously we had only tested the DNA samples for the alternate reference for Jessica Haringa and Rebecca Bletch using 16 locations. So I then just looked at them in a different system.
Okay. Anything different about that particular report than the original one that you drafted?
No, there's not.
I would move for the omission of People's Exhibit 182.
No objection, Your Honor.
Receipt.
We're going to stay in that same laboratory number, record 19.
Yes, sir.
It's 183.
183, yes.
I'm good.
Okay.
Thank you.
Thank you, sir. All right. Handing you what's been marked as People's Exhibit 183.
Is that a report that you have authored?
Yes, it is.
Okay. And again, is this a DNA analysis report?
Yes, it is.
Now, it does contain a couple of samples that you had previously testified to. Is that correct?
Yes, it does. It looks like there are 3 samples I've previously testified to.
Okay. So we have, in this particular report, you are looking at the swab of the exterior of the Reebok glove. Is that right?
That is correct.
And the swab of the end of the dildo vibrator, correct?
Yes, it is.
And then it looks like we've added some known references to the original list. Is that an accurate statement?
Yes, it is.
Okay. So let's look at your comparisons then to the first part of this, which is the swab of the end of the dildo vibrator. What were you able to, I guess, what comparison, if any, were you able to make?
The next I could then compare separately. So this was the swab of the end of the dildo vibrator. This was a mixture, like I previously testified to, a mixture from 2 individuals. I had previously compared the DNA profiles of Jeffrey Willis and Madison Nygaard, and I then made some additional comparisons to Rebecca Bletch, the alternate reference we had for Jessica Haringa, Kevin Bloom, Charlene Ann Willis, and Michelle Shinotola.
Okay, and in your comparison, your new comparison, I should say, what were you able to determine?
I first compared the DNA profile of Rebecca Bletch to that mixture from 2 individuals on the swab of the end of the dildo vibrator. And when I looked at that, I set up the scenarios to see if it, given the evidence, if Rebecca Bletch is a possible contributor, or given the evidence, is Rebecca Bletch not a possible contributor. So the 2 scenarios I set up were the DNA obtained from the swab end of the dildo vibrator is from Rebecca Bletch and 1 unknown unrelated person, or is that DNA obtained from the swab end of the dildo vibrator better is explained as originating from 2 unknown unrelated people, neither of whom are Rebecca Bletch. And based on the DNA results for the swab of the end of the dildo vibrator, it was at least 4,600 times more likely that it originated from Rebecca Bletch and one unknown, unrelated person compared to if it originated from two unknown, unrelated individuals. And that analysis and that calculated statistic provides strong support that Rebecca Bletch is a contributor to the DNA results obtained from the swab of the end of the dildo vibrator.
Now, you also made some exclusions, is that correct?
Yes, I did.
Okay, what were those?
I was able to exclude the alternate reference for Jessica Haringa, the DNA profile of Kevin Bloom, the DNA profile of Charlene Ann Willis, and the DNA profile of Michelle Shinatola. They are excluded as possible contributors to the DNA obtained from the swab of the end of the dildo vibrator.
All right. The other comparison was to the exterior of the Reebok glove, is that correct?
That is correct.
Okay. And what were you able to determine off of that? I guess your first comparison. Let's talk about your first comparison.
So the swab of the Reebok glove exterior was a mixture of DNA from two people. I first compared the DNA profile of Rebecca Bletch to that item. So the two scenarios I looked at to see which one better explained the evidence that was detected on that sample were the top part, or the first scenario I compared. The DNA from the swab of the Reebok glove exterior is from Rebecca Bletch and one unknown, unrelated person. Or is that DNA obtained from the swab of Reebok glove exterior better is explained as originating from two unknown, unrelated people? And based on the DNA results for the swab of the Reebok glove exterior, it is at least 90 quadrillion times more likely that it originated from Rebecca Bletch and one unknown, unrelated individual compared to if it originated from two unknown, unrelated individuals. And based on that analysis and calculated statistic, there is very strong support that Rebecca Bletch is a contributor to the DNA results obtained from the swab of the Reebok glove exterior. And for clarification's sake, the number 90 quadrillion is the number 90 followed by 15 zeros.
All right. The other comparison you made was to who?
The next comparison I made was to the DNA profile of Jeffrey Willis, to that mixture from two individuals. So I looked at the DNA profile of Jeffrey Willis and I compared the two scenarios. That first scenario I compared was the DNA obtained from the swab of the Reebok glove exterior is from Jeffrey Willis and one unknown, unrelated person. Or is that DNA better is explained as that DNA from the swab of the Reebok glove exterior better is explained as originating from two unknown, unrelated individuals. And based on the DNA results for the swab of the Reebok glove exterior, it was at least 47 million times more likely it originated from Jeffrey Willis and one unknown, unrelated person than if it originated from two unknown, unrelated people. And based on that analysis and calculated statistic, there is very strong support that Jeffrey Willis is a contributor to the mixture of DNA obtained from the swab of the Reebok glove exterior.
Okay. And again, in this particular comparison, you were able to you made some exclusions as well, is that correct?
Yes, I was. I was able to exclude the following people as possible contributors to the DNA obtained from the swab of the Reebok glove exterior. The following people are excluded: the alternate reference for Jessica Haringa, the DNA profile of Madison Nygaard, the DNA profile of Kevin Bloom, the DNA profile of Charlene Ann Willis, and the DNA profile of Michelle Shinatola are all excluded as possible contributors to that mixture of DNA obtained from the swab of the Reebok glove exterior.
Okay. And again, does that exhibit, People's Exhibit 183, is that I guess, exact copy, or is there anything different in that exhibit than the original report that you submitted?
No, there is not.
Okay. Your Honor, I move for the admission of People's 183.
No objection, Your Honor.
Received.
Oh, yes, sir. Okay. Last report that I have for you, Ms. Schmidt, is marked People's Exhibit 184. This is referencing GR 13-2559, Record 18. Okay. This report, again, are you using the StarMix program?
Yes, I am. If you'll just give me a moment to turn to my copy.
Absolutely. Let me know when you're ready. Set? Okay. In this particular record, we're talking about an analysis of what?
In this report, I am looking at the following DNA samples. The swab of the Carl Walther / Ulm battery cover. I'm looking at the DNA profile for the alternate reference for Jessica Herringa, the DNA profile of Kevin Bloom, and the DNA profile of Jeffrey Willis.
Okay. Now, were you able to determine whether or not you had a single source or multiple source in that initial analysis?
The DNA profile from the swab of the Carl Walther / Ulm battery cover was a mixture of DNA, and that was a mixture of DNA from 2 individuals.
Okay. And what hypothesis or what comparison did you make?
I first looked at the DNA profile of Jeffrey Willis to that mixture from 2 individuals. So the 2 scenarios that I compared in this case to see which one better explained the evidence that we were able to detect on that sample. So that first scenario I compared was the DNA obtained from the swab of the Carl Walther Ulm battery cover is from Jeffrey Willis and 1 unknown, unrelated individual, or is the DNA that was obtained from the swab of the Carl Walther Ulm battery cover better is explained as originating from 2 unknown, unrelated people. And based on the DNA results for the swab of the Carl Walther Ulm battery cover, it was at least 36 times more likely that it originated from Jeffrey Willis and 1 unknown, unrelated person compared to if it originated from 2 unknown, unrelated people. And based on that analysis and calculated statistic, the swab of the Carl Walther / Ulm battery cover with regards to Jeffrey Willis is uninformative, and I can make no more conclusions on that.
You had some exclusions as well?
Yes, I was able to exclude Jessica Herringa and Kevin Bloom as possible contributors to that DNA sample.
And I do notice that Jeffrey Willis is not on that list. Is that correct?
No, it was an uninformative comparison, or basically an inconclusive comparison when I compared the DNA profile of Jeffrey Willis to the DNA sample obtained from the Carl Walther / Ulm battery cover.
And you can't say he's excluded?
I cannot say he's excluded, correct.
This report, People's Exhibit 184, is there anything different from the original that you submitted in this particular case?
There is not.
Your Honor, I move for the admission of 184.
No objection, Your Honor.
I have no further questions of Ms. Schmidt.
Yeah, I'm sitting on there, maybe. We should let it go. That's training issue. That being the case, John, I'm going to have to get in there. Yeah, I got it. Thanks. Well, we'll see if I got it. All right. Good afternoon, Ms. Schmidt.
Good afternoon, sir.
We have, Mr. Kresnick, do we have a marker?
They're over here, Fred.
They're over there. Huh? Several markers, thank you. All right. Mr. Schmidt, we did this the last time, so I think you know what's coming here. Earlier today, your colleague testified. Let me find it here. Testified of the ratios and the likelihood my client's DNA being on a handgun. I'm trying to find the number that he used here. I think it was 6.24. Let's see. No. Give me a minute here. He did a lot of talking. Okay. 6.24 septillion. Do you know? You would know that number is correct. What a septillion is?
Yes, I do.
Would you please write 6.24 septillion on that board and with the zeros so we can see how many zeros that includes?
Sir, do you have a color choice that's better for everyone to see?
Go crazy.
Great. Could you repeat the number, please?
6.24 septillion.
Sorry, I did not leave myself enough room.
I'm sorry?
I did not leave. I started too large. I wrote too large to begin with.
It's okay, you don't have to put it on one line. Matter of fact, make it bigger so everyone can see how many zeros that are involved with that particular number. Can you all see that writing?
You like this color choice?
Thank you so much. So we're talking 3, 6, 9, 12, 15, 18, 21, 24 zeros,
20.
and before we get to the 240. So 25 zeros plus 624, correct?
So it would be moving the decimal point over basically 28 spaces.
Okay.
So
Oh, that's fine. As long as that's 6.24 septillion.
you said 6.24?
Yes, ma'am.
I believe that should be right. I may be off on 1 0.
We're not going to hold it. No one else in the room would know, so don't worry about it. Okay, that number represents more human beings that have ever existed on this planet, doesn't it?
I don't know an exact number of the number of human beings that have ever existed on planet Earth.
Okay. You know about, your predecessor says that there's somewhere between 7 and 8 billion people now, correct? Is that, you know that?
That's an approximation. I don't know what my coworker testified to the current population of the Earth.
Okay, but that's more than 7 or 8 billion, correct?
Yes, it is. A billion would have 9 zeros behind it. That I am sure of.
So a billion, this is 1 billion with this zero way back here, correct?
Yes, with 9 zeros
Okay.
preceding it.
All righty. And by the calculations that you've explained to us, I think it was your last exhibit was 18? I moved my page, so I missed Exhibit 184, your record number 18. You said that my client is 32 times more likely than other folks to be the donor of that DNA. Is that correct?
No, it was the number 36.
36, you're correct. Thank you. My apologies. 36. Okay. And, Ms. Joseph, would you be so kind as to scroll down to that chart on the bottom of this particular document? There you go. That's the one. You see that on your monitor there, Ms. Schmidt?
Yes, I do.
Okay. And the uninformative category peaks out at 99. Is that correct?
Yes, it is.
So this is just a little bit over a third of uninformative. Correct?
Yes, it is. I'd also like to clarify.
No,
that-
no, but I can ask you questions, but you can't. Well, he'll let you clarify. But on my turn, let's just answer my questions, okay? I don't have a lot of them. So, and the word that's used on the chart that you use is not inconclusive, it's uninformative. Is that correct?
Yes, the qualitative equivalent for the number 36 when comparing two different scenarios is in the uninformative range.
Okay.
Basically, the uninformative range is where I can't make a conclusion either way if that person could be a contributor or is not a contributor, which
So
is
you-
also inconclusive result. To that
So you've
comparison.
defined uninformative for us. You've defined what the word uninformative means?
that no information can be gained.
No, I'm asking, did you just define uninformative?
I believe so, yeah.
Okay, so, but I didn't ask you that, right? I didn't ask you to define the term. I just asked you what the chart said, correct?
Could you repeat the question?
I just, the chart said, I didn't ask you to define anything.
Could you repeat your previous question, please, sir?
What does the chart say? Does the chart say 0 to 99? What's the word the chart uses?
The word that the chart uses is uninformative.
Thank you. One moment, please. Okay. Right. Ma'am, have you used the phrase, a negative control associated with this case did not perform as expected? If I read that to you, would you know what I was talking about?
Yes, I would.
What does a negative control associated with this case did not perform as expected?
When we do all of our DNA testing, we have both positive controls where we have an expected result, so we know that every part of the test is working properly. And we have a negative control in which we should get no result. Some cases we do obtain possibly a single peak. We then show that data to our technical leader. We ask our technical leader to either approve So we could still use the data associated with that negative control, or we asked our technical leader to say we cannot use that data. So if we've put that in a report, our technical leader has reviewed the data that we've provided, showing the negative control that did not perform as expected. She approved that that data was still able to be used in casework.
Thank you. Ms. Schmidt, I have no further questions.
Just real brief follow-up. If you got a number 0, what does that mean?
If a for a likelihood ratio.
Yes, I'm sorry.
If when I performed a likelihood ratio statistic on a comparison of a piece of evidence to a known DNA profile, and the calculated statistic in that case was 0, that would be an exclusion. And in these cases where I did exclude individuals from the mixtures of DNA, all of the exclusions had a calculated likelihood ratio of 0.
Okay. So in order to be excluded, you have to have a 0, just so I understand that.
To be excluded from a sample, you would have a calculation of a likelihood ratio of 0. Correct.
Thank you. I have nothing else.
Judge, I have no further questions. Thank you, Ms.
Thank
Schmidt.
you, Ms. Schmidt. Can you stand down? I believe you indicated to me that was your last witness.
Yes, sir.
Okay, we're going to break early today then. It's 5 o'clock. You may stand down, Ms. Schmidt. Ladies and gentlemen,